Okay, let's get the ball rolling. So today's topic is going to be on FTM one, more specifically on week two and a little bit of week three. We didn't want to overwhelm you guys with a lot of week three stuff, so just be patient with us when because a lot of the things that you're going to be covering this week are going to be brought up today. We wanted to give you guys an early kind of preview of the material so that it's not like, oh man, I have to show up to lecture the next day and kind of be traumatized. So bear with us. We're going to go over some high yield concepts that are likely going to be tested based off of what we personally experienced, as well as what you will likely experience. You guys ready? Okay, let's get started. So, important thing, okay? You are going to get through this. There's just positive messages before we kind of go along, okay? We're a whole bunch of DES pages. We have Sparkles, Dog Mom, Scrubs, and here's the TPLG. All of them are dedicated to help you guys make it through the next, week and then the rest of the term as well. So just stay positive. Make sure you sign in to Turning Point because we have a warm up kind of question coming up. But make sure you guys participate because it's shown scientifically and statistically that those who do more practice questions and more hands-on experience of this material tend to do better on the exams. So feel free to also message if you're not able to use Turning Point, message in the chat your answers. Okay. Session IDs down below, just be aware of that. Okay. Let me quickly do an introduction. My name is Kishore. I am a term two student who made it out through term one. So um, I personally run with several of our hosts today, the Sparkles Gang DS page, and personally I run the Here's the TPLG as part of the kind of collaborative efforts between the various DS groups.
Can we go next? Okay. Hi, everybody. I'm Sarah. I'm also a T2. And then I did CFP before term one. I'm also part of the Sparkles Gang DES, and I run the Golden Ages DES group at the same time. And I also do OPLGs. So sign up and don't leave me alone.
Yep, and I am Cassidy. I am also part of the Sparkles Gang DS group. And I do not run any other pages on the side of that other than just AEP. I think I see some of my AEP people here, so that is good.
So that's all of us. We also have Lexi Luong in the chat. She's also managing Sparkles, so you can also message her and message us just any questions on the side about term one and everything. So just be aware that those are your hosts for today's meeting. Okay. All right. Let's do the next thing. So let's open up polling. Let's warm you guys up with a good, hopeful, practice questions. Okay. And I'll give you guys some time. Oops. Give me 1 second, guys. Sorry. Turning Point's having a day. As always, Turning Point always has its day when you least expect it. Give you guys some time.
So the PowerPoint should be posted on the Sparkles DES Facebook page. If you guys can hop on over to Facebook and just download that if you don't have it. And then if not, we'll post it on all the other pages. I think Kishore already did. Right?
I haven't posted on the other pages, didn't
Okay.
get a chance, but it will be posted in the other pages after the session.
Mhm. Yeah, sorry. Anyone that is joining right now, I am furiously adding people to the group. So just be patient with me. I will add you.
Okay, let's discuss it. So the answer the majority of you guys put is A. Good job, nicely done. Um, it's important that you remember that these factors, right? That you remember Chargaff's rule of having equal levels of adenine to thymine, guanine to cytosine. Of course, DNA, what's the charge on it?
Negatively charged.
Negatively charged, exactly. It's going to be important because you will likely encounter very soon about histones and methylation and all of those things. And it's going to be very important to remember that DNA itself has a negative charge, and the histones, being the positive charge of the family, are going to be able to nicely bind to them and help maintain kind of a condensed genetic structure. Okay. All right, so let's get started with this. This seems to be a concept that initially a lot of folks struggled with. It goes over specifically mRNA slicing. What is the first donor site, guys?
GU.
GU, exactly. GU is a very important one to remember because the donor site and the acceptor site are kind of universal in most cases, right? Whenever you see a donor site, the transcription factors are going to bind in and splice out that particular intron based off of the donor and the acceptor site. Okay, the acceptor site, what is its genetic composition? What's the two letters?
AG.
AG. Okay, really, really critical to remember those two. Commit it to memory because they will likely ask you questions on splicing. So as always, keep that aware. So why do we spend so much time learning about splicing? Because remember, our genetic information is synonymous across every cell of your body at this very moment. Obviously, you don't need your brain cells to be making something that a muscle cell is supposed to do. So all you can do in the long run as a cell is to do alternative splicing. Okay, that's just a short summary of it. Obviously, it's not the like end all be all of responses, but alternative splicing in this case typically tends to be the answer for most genetic diversity that you see within a species, within a organ, within what you see overall in your body, right? Your skin cells have the same genetic composition as your brain cells. So it's important to remember that alternative splicing varies from location to location and in terms of what protein or product is made and its various functions.
Sorry, Kishore, let me just interrupt you really quickly. Can you move the session ID over because I've gotten a lot of requests? Okay.
Sorry.
It's okay. Thank you.
No problem. Cool. Exactly. So donor and acceptor sites, you guys already said GU and AG. Okay. Obviously, you have to keep that in mind because when you're going through this, you're always going to be concerned about particularly where you're splicing, right? A cell that is in the liver is going to splice a little different than what is going to be spliced in the muscle tissue. Okay. Now, let's go to the next thing. Here's some examples of alternative splicing in its finest. These are obviously what type of modification, guys?
Transcription.
Transcription, exactly. It's transcriptional because remember, we haven't created a product yet. The product is going to be your amino acid sequence, but in this case, we're just making genetic level changes in your mRNA, right? We want to, what are we going to excise out? Are we going to excise out the introns or the exons?
Introns.
Introns, exactly. And sometimes we actually take out a bit of the exons because remember, once again, the key concept, alternative splicing in this case. So here's a great example of alternative splicing. You can see one of the most highly conserved one is the alpha tropomyosin gene, right? But look, if you see here, your striated muscles have a different composition of mRNA for the exact same alpha tropomyosin as compared to what you're going to see in the smooth muscle mRNA. That key principle, if you can commit it to memory, shows you why we have so much genetic diversity, right? In between cells. Okay. Please commit this one to memory. I swear we had a question on this before. It was the apolipoprotein B in the human liver. It's very much in the hepatocytes, it's different as compared to the enterocytes, right? We can see the composition of these in various degrees. Let me do it this way. A second. Okay. Sorry, I'm having a bit of technical difficulty. Okay, let's do a question on it where I explain it a little further. Give me 1 second. Let me open up polling. And we have 5 questions. Make sure everyone clicks in. Give you guys another couple of minutes. Okay, let's stop polling. Let's see how you guys did. So you guys picked C. So let's look at it. And you guys are correct. So the answer is C. Now, does someone want to take this opportunity to explain why you guys picked C?
I could do it.
Oh, okay.
Or Rochelle, go for it if you want.
All right, thank you. So the donor site and the acceptor sites, that would be like around the intron. So for if the mutation's on intron number 1, the donor site would be like to the left of the word intron 1, and then the acceptor site would be to the right. So if there's a mutation in the donor site, then nothing would be cut to the left of the word intron 1. So intron 1 would stay in. So the next following donor site would be the one for intron 2. So intron 2 would be cut out, but nothing else would be.
Awesome. That was a perfect explanation. I love that. You explained it very thoroughly. Exactly. So keep that in mind, guys. In terms of splicing, they can present questions like this in terms of like you can have like a weak acceptor site, a weak donor site, and it can really tell you where you're going to have the splicing. It's if you can get that question down very well, then you're going to be a-okay. Okay. Awesome. Let's do this question. Give me 1 second. Let me open up polling. Awesome. I love that you guys are participating in the chat and looking at other ways you could be tested on this because They might not give you the exact same example like the one they gave in lectures. So just be aware that if there is a mutation in the acceptor site, it will likely jump to the next possible acceptor site based off of what the composition of the GU to AG is. Okay. Make sure you guys click in. And that's time. So let's see how you guys did. You guys picked C again, and that is absolutely correct. So what is this question asking? The question is asking if the RNA below has a mutation in the splice acceptor site for intron 1, what would the structure of the mature mRNA going to be? So is the donor site affected in this case?
No.
No. So if the donor site is conserved and everything is okay, what we're likely going to see is a issue with the next possible site, which is going to be AG, right? So that means which portion gets spliced out, which exon gets spliced out?
E2.
Exon 2.
Exon 2, exactly. Keep that in mind because it's going to be very critical that you remember. They might not ask you in terms of just a linear sequence of like, oh, like, just tell me what the end product is. They might ask you in terms of what gets spliced out. That's just another way that they can test your knowledge of your donor and acceptor sites. So why E2? Okay, that was a question in the chat. So why E2 gets spliced out? Because remember the AG site that's here gets removed, and that removal causes a bit of a problem, right? Not removed, but there's a mutation on it. So the splicing factors aren't able to see selectively the first AG. So they have to go to the next possible AG, which then allows for E2 to be excised out, leaving only E1 and E2, or sorry, E1 and E3. My apologies. Okay. Question.
Yeah. Hi, Kishore. So if it was a donor site, it would just be the intron 1 that would be spliced out, right? On this example here.
If it was just donor 1 that was having issue, is that what was the question?
Yeah. So if it was just the GT site that was mutated, then it would just be the intron 1 would stay in, right, at the final product.
Correct. It would. Because remember, what gets excised out would then be the next donor site. So it'll be the intron 2, which is the little arrow after E2, that portion, and AG would get spliced out. So what your new sequence would be is E1, intron 1, E2, and then E3.
Thank you.
Yep, no problem.
And just a clarification again, just to make sure I have this concept down. But because an acceptor site was mutated, you have to skip to the next acceptor site. But before you can get to the next acceptor site, that exon that was next to the mutated acceptor site has to go along with it.
Yeah, it's kind of
Okay.
like it's kind of like a skip, right? It's like looking for the next possible location that it can accept. Because remember, once the donor site is selected, it's going to look for the next possible viable acceptor site. That's why the E2 exon 2 gets removed out.
Yeah, so when you're thinking about it, the way I looked at it when I was doing these problems is your splicing is going to go in sequential order. It's always going to be donor acceptor. So if your donor's messed up, it's going to look for the next donor. If your acceptor is the one that's messed up, you're going to splice that first donor and look for the next possible acceptor. So pretty much if they tell you acceptor site is messed up or mutated, look for the next possible acceptor site. And that's like how you know what's being spliced out. So if your mRNA is getting longer or shorter.
Did that clarify anything? Any follow-up questions on this? Because this tends to be like, oh, it's simple. Like you go to the next site, you grab the next thing and you move on with your life. it can be a little bit tricky, so feel free to ask your questions. I'll give you guys a couple of seconds.
I have a quick question,
Go for
that's
it.
okay.
Yeah.
I remember in class he said something about like if the donor is being mutated, the thing would be longer, and if the other one's being, it would be shorter. So just so I could write it down as on the side, which one would result in a longer, which one would result in a shorter? And is that the protein that's being longer or shorter, or the RNA that's being longer or shorter, if that makes sense?
Sarah, want to take it?
Yeah. Okay, so for this one, let's use this one as an example. Let's say we're telling you that there is a mutation at your first donor site, so that first GT after exon 1. Then your splicing is going to, sorry, let me see if I can get like a laser pointer or something. Oh, I can annotate and then I'll just delete it. Okay, so let's say this is where your mutation is, then you're not going to splice at this site because it's never going to splice at acceptor site first. So you're going to splice here. And then your next acceptor site would be here. So this is the only section that I am splicing out. And that means that this part is being included in my mature mRNA sequence. So would that make my mRNA sequence longer or shorter? Anyone? You guys can unmute yourself.
Longer.
Longer, exactly. But if you look at it the other way, and now we're talking about a mutation in this acceptor site. So I spliced here. I'm looking for an acceptor site. I can't splice here because there's a mutation. So I come to this one over here, and this is a donor site. So I'm not going to splice here because I'm looking for an acceptor site. So my next possible splice is going to be here. So I am splicing out this entire section. And now is my mRNA sequence going to be longer or shorter?
Shorter.
Shorter. So does that make sense? For whoever
Yes.
asked the question?
Yes, yes.
Okay, so that's kind of like what you want to have in the back of your head. So I wouldn't go as to like memorize this concept particularly. I would just try to understand it, because no matter what question they give you, you can figure it out based on if they're saying this is the mutation, this is not, like this is the mutation. So you want to be able to figure out what the answer is no matter what the possibilities are. Another one that they sometimes put in is a splice site right here. Like let's say they say, oh, I added in a acceptor splice site at this location. So then I cut my donor site here. I cut this new acceptor site because it's the first possible acceptor site. And so now I'm looking for a donor site. So I completely skip this guy over here. And that would make my mRNA longer, but it's not going to include this entire intron like the other ones because this is still being spliced out. So I hope that makes sense to you guys. If you have any other questions, we can go over it again at the end, but I hope that made sense.
Sarah, just a quick question, if that's okay.
Uh-huh.
So the example you just gave right now, if you put the splice site right in between the intron in the middle, so the second portion of the intron will still be included, right, in the final product?
Yeah, exactly. Because I can't splice two acceptor sites right after each other. And
Mhm.
since there's no donor being added also, then I'm going to end up with a little bit of intron in my mature mRNA where I was supposed to have no intron.
Okay, thank you.
No problem. Sorry, Kishore, go ahead.
No, this is perfect. That was you explained it very thoroughly. Okay. Keep that in mind. You don't need to have it right after a exon, right? You could have it potentially midway through a intron. There are all sorts of genetic variations and things. And that was the question that one of you guys brought up. So just be aware of that too, in terms of 1/2 of the intron could be conserved, whereas the rest of it could be excised out. Okay, we're now going to move into pedigrees. And if you guys are still wobbly on this topic, we will, like Sarah said, come back to it. So All right, so pedigrees. This seems to be affecting a lot of folks in terms of like what the heck is going on, in terms of why are we looking at this? Why is it so daunting? Here is the nomenclature that you need to have kind of roughly down. They're not going to present you all of it because they don't expect you guys to become geneticists, unless that's your jam. Feel free to do that. The pedigrees here, they have a particular kind of system that they follow. If you see like consanguinity, it's double lines, okay? If you see someone affected, it's gonna be a filled in box. The males are squares, females are round, okay? That's just the basic ones you need to be aware of. Obviously, like if you see death, it's just a crossed off box. It's not filled in or anything, it's just crossed off typically. And then heterozygous status is the one that's typically the less commonly seen, but they can give it to you. And that's half of the box being filled in, okay? Those are the basic ones to keep in mind. The other ones you will likely not see with the monozygotic twins and dizygotic twins and everything like that. So just be aware as to which ones you're likely going to see, right? Based off of this. Obviously you guys know mating because you guys would have seen that in lecture, and then affected. Death is an unlikely one, and then heterozygous is also an unlikely one, because most of the times they can ask you test questions based off of the heterozygous status of things. They tend to not give you that one, so just be aware of that.
There is
Right.
no difference between heterozygous and carrier recessive ones, right? Because for being a recessive carrier, you have to be heterozygous.
I believe so. Sarah, unless you've heard differently.
Yeah, so heterozygous could mean that it's a heterozygous carrier. It could also mean that someone is heterozygous for a dominant mutation disorder. Sorry, I can't think. But they usually don't use heterozygous for us. They'll use this little dot for recessive, like carriers. And then for autosomal dominant disorders, they usually just fill in the whole thing to show that that person is affected. So this one is kind of less used, but just assume that that person is heterozygous for whatever mutation you're talking about. I hope that clarified it for you.
Sarah, do you want to do the autosomal dominant ones?
I think Cassidy's doing right.
I'll take this. Just to clear that up though, so sometimes like if you guys have to, they definitely don't give us the heterozygous, like the half box thing, because if you think about it, sometimes like if people are heterozygous for an autosomal dominant disorder, they're going to be affected. So they're going to be actually filled in. So just try not to think of it as being heterozygous or homozygous as far as being filled in. If the box or the square or the circle is filled in, then that means that they're affected. So try to think of it that way. So like, first of all, autosomal dominant. These are going to be, you'll see if someone is affected, autosomal dominant, you're going to see it in every generation. And that's what this means by vertical inheritance here. So you can see here, there's one person in each generation, and you guys definitely get familiar with your pedigree, like nomenclature and all that stuff. So like the 1, 2, and 3, each line is going to be a generation. So each generation here, you see somebody that's affected. The males and females are typically affected with equal frequency, and you do see male to male transmission. So that's how you know that this is going to be autosomal dominant and not like an X-linked or something different. Okay, so this is kind of more the science behind it, the Punnett square. So you can see here, if you have a normal parent that's not going to have the allele, then you can, if that person mates with an affected parent, the affected parent is heterozygous. So you have an affected parent and a normal parent, and there's going to be a 50% chance that any of their children, regardless of the sex of them, are going to be affected. So it's each event is independent. Each childbirth, like you could have 4 kids with the disease or 4 kids without the disease. They're all independent of one another. Okay, here's another pedigree. This is just giving another example so that you guys can see. You can notice here that there is at least one person in each generation that's affected, and then that you're also seeing about an equal number of males and females affected. Okay, so I'm not going to go super into the details behind each of these disorders for any of the inheritance patterns, but I want to make note that it's very important to go ahead and get these down now, because the thing about genetics is, especially with these disorders, they're going to continue to come back, like not just in FTM, like MSK, CPR. We just got asked about some of these on our exam this past week. So like learn everything you can about these early and try to keep up with it, because it'll help you a lot in the future. Like the genes that they're associated with, the mutations, the symptoms. The cool thing about genetics, in my opinion, is they're kind of buzzwordy as far as the symptoms. So like if you have a slide that's, you know, describing Marfan syndrome, and you are given words like you know, the person's like taller than normal and they have long limbs, long fingers, or like the barrel chest, things like that. Go ahead and remember those things, because if you memorize those type buzzwords as symptoms, you'll be able to pick it out right away when you're reading a vignette on an exam question, and it'll help you a lot. And you can almost to the point where like you don't even necessarily need the pedigree sometimes to answer the question, and you can help yourself out just by going ahead and like memorizing all of that. So I would definitely recommend that. For the AD ones, you can use the mnemonic Man of Ham, and that just gives you the first letter of all those there. So that's what that's about.
Can I ask a quick question regarding these?
Yes, of course.
Do we have to memorize the specific pathway that these diseases are caused by? Like, let's say.
Some are more important than others, I would say, but like if Dr. Sobering gives you the gene, or especially the gene that causes the that has the mutation, or the type of mutation, or the things that go with it, like I'm gonna get into those too, but like the gain of function versus loss of heterozygosity versus things like that, like all of that. Yeah, it's probably a good thing. You probably like, it's good to just get to know these really well from the get-go, because it's going to continue to keep coming back. It'll come back more on your FTM2 exam, and then it will come back more and more throughout the term as you learn more about each of the diseases, some more than others. But yeah, I would recommend knowing anything that Dr. Sobering tells you to know. That's why there's so many DLAs on these, because there's a lot of information, but it is really important.
And a lot of times, just to quickly build on that, is you're going to see a lot of times where they might not even give you the disorder. They might just tell you the gene. They can be like Marfan's, fibrillin 1. And then you have to know what are the follow-up like pathologies and things that is associated with it, so that you can make that quick like snap decision of saying, okay, we're talking about Marfan's here. What is the inheritance pattern? Autosomal dominant. They won't ask you to become clinicians overnight, where they'd be like, oh, like Marfan's, it's going to present with very long limbs. They might give you that in the question stem, but most likely at your guys' level, they're going to ask you questions on like, if it's, you know, fibrillin 1 pathology, what is the inheritance pattern for this disease? Or they might ask you something along the lines of, if it's Marfan's, what is another condition in the autosomal dominant category that also fits into this? So those are just different ways they can likely test you on this material.
Yeah, I agree with that. I would actually go as far as to say, like, most of the time they're not going to give you the disorder. This is like, genetics is a really good area to ask like second and third order questions, where they might just describe somebody and some symptoms that they're having. And then they're not going to ask you like what the diagnosis is. They're going to say, like Kishore said, what is, you know, another disease that has this type of inheritance pattern, or what might this person be at more risk for, or would this be an example of like gain of function, or, you know, heterozygosity, like all that. So yeah, genetics is very important. So it's good to just like know everything. If Dr. Sobering told you to know it, then just know it, because it's going to be important for like the whole term. Okay, good there. Okay, same thing with autosomal recessive. I honestly think these are probably more important, because there's a lot more to this, and there's like math and stuff. So this is going to be horizontal inheritance. This means that there does not need to be a person in each generation that's affected by this. And the reason for that is you need two parents that are carriers to have a child that's affected. And that's because you are going to be homozygous for the disease-causing allele in order to be affected. So it's going to be less people affected than in autosomal dominant, and there's not going to be someone in every generation in these. Same thing with the equal frequency with the males and females, though, for autosomal. Okay, Kishore. Okay, so here's like the visual of it. This would be, you have two people that are carriers, and they're gonna have 75% phenotypically normal children. 25% of them are gonna be affected because you have to have both disease-causing alleles to be affected. But what's more important, actually, go ahead, Kishore, and go to the next one, is this 2/3 rule. This gets tested a lot. And I know you guys have gone over this because Dr. Sebring's good about giving a lot of practice questions on his DLAs and lectures. I actually noticed that when I was going back looking through, do all of those like multiple times, because they try to trick you with these, and you don't have like anything to write down during your exam. So you have to like get in the habit of knowing if they're asking about a sibling being a carrier, you need to know the 2/3 rule. And the reason for that is they might ask you about an aunt or an uncle. So it might go a generation out, and they most likely will, because it's a little bit more difficult. So you have to think about like the mother of the child and then their sibling. And so that's when you have to like do a little bit of math in your head, and it gets to be a little bit difficult. But you have to know the biggest thing about this, like the thing that took me a little bit to understand, is that the person, if they were affected, then the carrier status wouldn't be in question. So if a family has a child and they're trying to figure out if the child is a carrier of the allele, they're going to not count that homozygous recessive, because they're going to assume that the child is not affected. And this is only applicable to the disorders that are penetrant at an early age. So like ones that you would know when your kid is a baby, but that's where the 2/3 comes in. So there's going to be a 1/3 chance that they're homozygous normal, and then there's going to be that extra 2/3 that they are a carrier for the disease causing allele. So know this super well. I think there's a practice question on this, but yeah, you have to know this. This is going to get asked more than once, and probably like throughout the entire term.
So in the chat, we're getting a lot of people asking about the 2/3 rule. That's exactly what Cassidy just explained. So like she said, if you have a child that's affected, you're going to know that that child is affected. They're showing all the phenotypic symptoms. So when you're trying to figure out if their siblings are carriers, because you know they're not affected, you just want to know if they carry that one copy of that mutation. So you cancel out affected because they're already born, you know there's nothing wrong with them. So you cancel out the homozygous recessive. And then you have a 2/3 chance that they are carriers and a 1/3 chance that they're completely fine and they have no mutations. So Cassidy just explained it. I just explained it. I really hope that makes sense. I know it's tricky for some people to understand.
Yeah, and the way that they'll usually ask this, well, a common way to ask this is like they'll say a child's mother, or a child has a maternal uncle that is affected with this disease. Then what is the chance that the child will have the disease, or what is the chance that the child's mother will pass the disease to child? So you have to think about like the siblings in the other generations of the family usually, and it makes sense. I mean, yeah, you just have to do a lot of practice problems on these, because that's the only way. The more you see it and the more you see like the terminology and stuff like that, you'll be able to get it a lot quicker. But yes, for those of you that are asking, it is only applicable for the autosomal recessive disease pattern. Yeah, it wouldn't be autosomal dominant or anything like that.
Can I ask a quick question, Cassidy?
Sure.
So when the example you just gave about the kid's uncle being affected, so then that would mean the mom is a carrier, obviously, because that would just be. So what kind of question would they ask on that? Be like, if the mom had a 2/3 chance, but she is a carrier for sure because we know the kid is affected, right? And
So yeah, well, usually there's going to be a number of different ways, but for example, they might not tell you, like, let's say a kid is born, they're not going to tell you if the kid's affected or not. They're not going to tell you if the mom is affected or not. They'll tell you that the kid has a maternal uncle that is affected. Okay, so then in your head, you have to think the mom is going to have a 2/3 chance of being a carrier. And like, maybe they'll tell you the mom is not affected. Okay, so then you'll have to actually do a little bit of multiplication, because it'll say, what's the chance that the child is affected by the disorder? And then it's usually going to be like your 2/3 rule times 1/4, because there's a 1/4 chance that the child will end up being affected based off of the squares, because you have to be homozygous for the disease causing allele. So that was pretty common, I think. They know that you don't have whiteboards and you can't write stuff down. So I think for us, we were the first term that took this course online, and they kind of, they said they took that into consideration and made the math easier. So I don't think they're going to go any farther than that, because you can't write it down, but you will have to do like one step of multiplication sometimes.
So would that be like a 1/6 chance? Because 2/3 times, I don't know if I'm doing my math right, but I know they mentioned something in the lecture where it would be 1/3, like 1/3 possibility for that child. If your mom's a 2/3 possibility of being a carrier, then that their child is a 1/3 possibility of being affected, right? Or something like that.
It would be yeah. So like just based off of any independent event, there's a 1/4 chance that the child will be affected, right? Just based off of like, look at the Punnett square, there's a 1/4 chance that the child will be affected. So you could do 2/3 times 1/4, and then that would give you the answer. So yeah, it'd be like, whatever that is, 1/6, I think. But yes, if you, just do practice problems with them, because it'll help you a lot, but there will be questions on this for sure.
Thank you.
You're welcome.
Can I ask a question?
Yeah.
What if in the same scenario, they would ask that, the uncle is affected, and then what are the chances that the child will be a carrier? Then would you multiply it by another 2/3, or would you multiply it by half?
I'm pretty sure that came up a lot in our term, and I want to say that Dr. Sobering said he wouldn't do that, but I don't know. Don't quote me on that. I'm not sure. I know that once it gets really complicated though, math-wise, I don't think they're going to make you do a whole lot. I would say you probably will need to do like one multiplication problem with fractions, and they won't make it any farther out than that.
And the mother would have to be an obligate carrier for it to even work. So if she's not a carrier, then the whole question doesn't make sense.
Right. Exactly. Good. Okay.
Can I add something here? I'm sorry.
Sure. Yeah, of course.
Yes, Dr. Sobering said in his last review that we can memorize this for now, that for sure if he's asking the child being affected is 2/3, and if he's asking you if the child is a carrier, means he's homozygous, is 1/3. That's why we arrive to 0.33 for being a carrier, and we arrive at 0.67, which is 2/3 for being affected.
For affected, we just showed it on the pedigree. It's not affected, because if you think about it, we're talking about probability. So you only have 4 possible outcomes. 2 of those are going to be carriers. So if you look at the math, like, we're just completely throwing out the purple box because the child's already born. So we know that this child is not affected. And that's why we threw out the box. So instead of making your chances out of 4, your chances are only going to be out of 3. And we're just trying to figure out, is this child a carrier, or is the child completely normal for both of the alleles? So with the 2/3 rule, the 67%, that's talking about carrier status in someone that's already born.
They can ask these a number of ways. That's why I'd recommend just doing a lot of practice problems, and I'll see if I can dig some up and we can send them out. But you will get further into this stuff when you get to population genetics, which is not right now. So just for right now, worry about, if Dr. Sobering says it, then yes, take that to heart, okay? Because he rules over we do in genetics for sure.
And I highly suggest, trust me, all 3 of us attended his office hours diligently throughout FTM. So, and going forward too, he will explain each and every single concept. If you need the mathematical explanation, he will do it for you in his whiteboard. So I highly recommend, if you still have follow-up questions after this review, attend his office hours. We are still happy to give you guys plenty of practice problems. There are practice problems within this, slides themselves. But if you need more, there are plenty of external resources that we can send you guys.
For sure. And he made videos for us and posted them, just because people, if you shoot him an email and you have a question about something, he will make a whole video and post it for you guys, and then you can go back and watch it a bunch of times. So yes, he's very helpful and he's very smart. So I would definitely recommend emailing him if you have any more questions.
And if you guys have any clarification questions on what we just went over, we'll go over it again at the end if you guys still need to clarify.
Sure. I think there's some practice problems with it anyways. So yes, For sure. Okay, so here's just another example of some pedigrees that are showing the disorders. On the left here, you'll see there's only one person affected in this family. On the right, you have, so as you'll see, there's like the consanguineous relationship, and you guys probably did talk about this, that like increases your risk of mutations and diseases and things like that. So just keep that in mind. Did you give me, hang on. It's not working. Okay, this same thing as with the dominant ones. Know these. Just know these. Chap gets cash. That's your mnemonic for this one. These I would maybe even say are more important than the dominant ones because they love to test these just because there's so many ways to ask questions about them. And a lot of these, like we just talked about some of these in our module in ER. So these will continue to come back as well. They're important. Know everything about them if you can.
Same principle too. Make sure you guys remember the mutations or the genes that were attached in your DLA, because they will present vignettes or cases based off of that, rather than just giving you like, oh, you have someone with cystic fibrosis. Now tell me what's wrong with them. It's never like that. It's always, once again, if you have cystic fibrosis, what is another pathology that's within the same broad category of autosomal recessive? And they'll give you like, you know, A through H or whatever.
Yep, I totally agree. And in my opinion, the questions that come from genetics are a little bit trickier, like Dr. Sobering, a little bit trickier, not as straightforward. So if you know all the details about these, then you'll be better off. Oops, sorry. Okay, here's a practice question for y'all, actually. Kishore will probably start the polling for you momentarily.
Polling is open.
Also in the chat, yes, I believe the A is for alkaptonuria, not albinism. So just double check with your notes, but
there's two different versions. So I think they both count actually, but yes, I saw both and I included
albinism.
Yeah, I'm not going to call anybody out, but anyways, I got these from from Brady. But
turning point, that's where we're answering these questions. Oh, Kishore, do you have that session ID? Okay. There it is.
I don't even remember going over alkaptonuria or whatever that is. I don't think that was super important.
It was one of the disorders that had like very little, like it was just a kind of like, I think the main thing was it was loss of function. So they didn't really focus on it for us, but I don't know if they focused on it for them. I just have it on my
That's all they said.
Yeah.
Just a loss of function, that's it.
Yeah, that's pretty much all we had to. Like, I have just like the summary table of all the disorders, and for alkaptonuria, that's literally the only thing I have written down: autosomal recessive, loss of function.
Okay, give you guys a minute and 30, a little extra. So I did flash up the answer. I do apologize. But yes, if you guys did the Punnett square for this, you would have been able to calculate it out. What are the chances of the child being a carrier? What is it, guys? What's the thing that Cassidy just talked about?
2/3 rule.
2/3 rule. So.
Very good. Okay, so with the X-links, they're different. These for me, I felt like these were harder to understand just because the X-linked, like you're gonna think females, but the diseases manifest themselves more severely in males and in higher frequency in males. And that's just because, actually I'll get to that on the next slide, but skip generations are common because this is recessive. So this is gonna be similar to autosomal recessive in that area. And it's not possible for male to male transmission. So a father can't transmit an X-linked recessive disorder because a male is going to be affected if they get one copy of the disease allele. And since it's going to be on the X chromosome, it's going to come from the mother. So there's no way that a male can give the disease to another male because they're not contributing an X chromosome. Now, a male can give the chromosome to a daughter, but it's going to make the daughter a carrier. They're usually, because they're going to be heterozygous, because there's only one X chromosome coming from the male. So that's why the X-linked recessive works out that way. I have a better explanation on the next slide. Okay, so yeah, this comes from y'all's slides as well. But this, for me, like makes it a little bit easier to see. So you only are going to get one X chromosome if you're a male in general. So if that one X chromosome that you get is the diseased one, then you're automatically going to be affected. With the female, you can get one affected X chromosome and still be like not affected. You can still be normal, but you're going to be a carrier. But then if you get two copies of it, then you will be affected as a female. And these are pretty 50-50 all around the board, I believe. So female to female transmission, there's a 50% chance that they're going to be, if the mom is affected, there's like a 50% chance that the female will be a carrier and a 50% that they will be affected. Or is it unaffected? Yeah, affected. And then with the male, there's just a 50% chance in general that the male will be affected if the mother's a carrier.
And just to go back to what you were saying about the females, that's what make them an obligate carrier, correct? Because either way, they'll carry it, but they have to have two copies to be affected. But men are always affected regardless because of the one X. Okay.
Yes, exactly. So that's just why there's more of a prevalence of males being affected. And then it's just keep in mind that it is also going to be more severe in males than it would be in females, even if the female was affected. So this is showing you kind of how it works on the pedigree. Here's those dots that we were talking about. So the dots here are kind of an exception. They're showing that the female is a carrier. So the female is heterozygous for it. And you can see that the heterozygous female, like for example, over here on the right side, to the pedigree, I know y'all can't see my cursor, but the right where there's an affected female, a normal male, they have one affected son, and then they have a daughter that's a carrier, but then they also have one unaffected son. So that's that 50-50 going on there. And you'll notice that there's 0 male to male transmission. So when there's an affected male, like on the left side, there's 0 male to male transmission. So he has 2 normal boys, and then he has one daughter that's a carrier for the disease. Okay. So here's what X-linked recessive would look more like. Try not to get it in your head that, just because it's X-linked, you're going to see always an affected female when you see an affected male, because more times than not, the mother might be a carrier. So the mother passed it on to her son, but she was never affected in the first place. So when they're not giving you that heterozygous state, you're going to basically assume that. So in this very first generation at the top, the mother would have to be a carrier because one of her sons has the disorder. But you're not going to see that. They're not going to always tell you that. They won't put that dot there, have the circle half filled in or anything like that.
So for this one, for the mother to be affected, for a female child to be affected, both parents must have the disease, right? Because otherwise the female would just be safe.
Yes, the female would have to get both copies from the mother. So I believe if the mother is affected, then she can have an affected daughter. But more times than not, the mother's a carrier because this is a recessive disorder. It's different when we get to X-linked dominant, which will be next, but yes.
So the father and the mother both have to be affected for X-linked recessive?
Yes.
Or you would have the one where the X chromosomes is not non-disjunction, right?
Yeah, exactly. So like with your X-linked recessives, if a daughter is affected, that means her father was affected and her mother was either a carrier or affected, but she has to get 2 of those X's one from each parent. So yeah, you're right. With recessive, it's like that. With dominant, that's where you start to see more girls that are affected, but we're going to get into that right now.
Yep. Okay.
You can have just the mom affected as well, right? For the daughter to be a carrier.
For the daughter to be a carrier, yes. Yes. Okay, so these are the X-linked recessive disorders. I would say that every single one of these is pretty important. Like we probably got tested over all of these, especially those muscular dystrophies. So just know these. Again, there's not really a mnemonic here for these, but just know them.
What's the first one? I kind of seem to forget about that one. How do you pronounce it?
Oh, the Lesch-Nyhan syndrome.
Yes.
Y'all want to help me out on that? I don't know off the top of my head which one that is. What were the buzzwords for that one?
Isn't that the hypoxanthia?
Yeah, so that's
Gout.
the PRT deficiency.
Oh,
Yeah,
yeah.
HGPRT. Mm-hmm.
Yeah.
Oh yeah,
And
y'all will.
there's a mnemonic for that one.
There is, yeah. I just was.
It's inappropriate, and I know this is being recorded, so I'm not going to say it.
You can put it in the chat.
Thank you for that. But with your Lesch-Nyhan, you're gonna go into a lot more detail when you do your purine pyrimidine synthesis stuff. So for now, just kind of remember what they're talking about, but usually they bring that up so that they can bring it up again in a later module. So
yeah,
don't worry
about
too much about it.
That definitely will come back. The HGPRT thing, yeah, for sure. Okay. All right, so X-linked dominant, these are more rare and there's only a few of them that you guys really need to know. But this is the one that you can have, the X inactivation. But the way this works is it's like a dominant one where it's not common that there's going to be skipping of generation. So there will be likely someone affected in every generation. There is a preponderance of females compared to males, and there's no male to male transition. But the reason that there's a preponderance of females in this one is because often it's lethal if a male is affected. So a lot of times they won't even make it to birth. And if they do, they'll die at a really young age. So you can go to the next one, Kishore. It shows you here that slash that we looked at in the beginning, that's showing you that that person, oh, wait, what did I do? Did I do that? Okay, so yeah, this one on the left here, you'll see that the mother was affected. She passed it on to one of her sons and one of her daughters. The daughter passed it on to a son who also died. So one of the original mother's sons died either in utero or at a very young age, or maybe they didn't make it to full term. And then same thing down here with this female. And then that's why there's a preponderance of females, because the severity is much worse in males. So often they don't make it, they usually will not make it to childbearing age in the first place, or child, what's that called? Yeah.
Yeah.
Yeah. So that's why there is that. And there's only 3 that are pretty important for y'all to know here. And that is on the next slide. Okay, Rett syndrome. Okay, so the first 2, Rett syndrome and incontinentia pigmenti, that's lethal in males. So Rett syndrome is something that you guys will talk about a little bit more. It's like a female with male-ish characteristics, but males do not make it to term, so they die in utero, and same thing with the incontinentia. Now, the vitamin D resistant rickets, I believe males can be born with and like live with, but they are gonna probably die at a younger age. And I know I mentioned on the last slide the X inactivation thing. That does come up a little bit more. Make sure you guys know like the X inactivation and how that relates to severity of the disease sometimes. And it kind of goes along with variable expressivity, which I have more on. But yeah, the exceptions like that are pretty important, because anything that's an exception to a rule, historically, SGU really likes exceptions and they like to test exceptions. So you're better off knowing things that are exceptions to a common rule than knowing the whole rule and like the normal stuff. So just a little tip there. Genetics is an area that they will get you with that as well. All right, that's that one.
To explain how X inactivation works with the X-linked dominant, is it that like one of the X's just gets inactivated, so that's why they're not going to be as affected as males? Like the bad X kind of gets inactivated. Is that what it means, or no?
Yeah, so the functional copy of it, of the X, remember females have 2 X's, right? So one of them turns into a bar body, okay? That's X. Depending on which X has the mutation or which X doesn't, it's going to show with the various level of like severity overall. So if it's the X with the mutation, obviously it's going to be more severe. If it's, you know, with the X that isn't doesn't have that severe of a mutation. So once again, you're going to have variable expressivity and all those, different terminologies we're going to cover in a bit.
Okay, thank you.
Okay, so
And this-
mitochondrial, oh, yes.
And this only happens in the X chromosomes, right? Not the autosomal, because even though you have 2 copies, you need both of them for expression, right?
Yes.
Yeah.
Yep. Okay, so mitochondrial, in my opinion, is one of the easier ones, just because like when I look at a pedigree, any kind of pedigree, the first thing I do is rule out mitochondrial because it's the easiest one to see on a pedigree, because the way it works is they're only passed from females. So the mitochondrial DNA that has the mutations will only be passed from the female, and a female is going to pass it on to all of her children. So if you see a circle in a pedigree and you see that all of the children are affected, you can assume mitochondrial. And then you'll see here as well, so the mom has 3 kids. She's got 2 girls and a boy. They're all affected. The boy does not pass it on to any of his children, and the 2 girls pass it on to both of their children. So that's showing you another generation of how that works. Okay, this heteroplasmy is seen in mitochondrial disorders. And what that is the severity of the disease is based off of how many mitochondria are involved. Basically, I was like reading up on this earlier because I forgot exactly how it worked, but the mother can pass either a little bit of the mutant DNA or a lot of it. And it can be in like varying degrees. You can see here, there's 5 different ones here. So the homoplasmy in the first one is going to show 0 with the affected DNA. And then the severest one will be, severest, is that a word? Will be the one that has the most mitochondria affected with the mutant DNA. So you can see a number of severity levels in these disorders, but just remember that they're always going to be passed from the mother.
A quick question for the mitochondrial diseases. The mother, if she's affected, like if she has some kind of mutation in the mitochondria, it would be like a circled in, it would be affected, right? She can't be like a carrier or something. Okay, thank you.
Yeah, Sarah and I were talking about this earlier. Yeah, she should be affected. I went and looked at it. So here is another example of this. This is why I rule these out first, because if you look at one, you should be able to tell pretty quickly if it's a mitochondrial. So you're looking at the mother here. She passed on the disorder to both of her children. And the girl also passed on the disorder to all of her children, whereas the boy on the other side did not pass on to any of his children. And to me, that's almost more important, noticing that the male did not pass on any at all, because you'll look, there's no further disorder in that generation. And then on the right side, you have the female that had 3 children. She passed it on to all 3 of them. And then the same thing goes a generation further. So the boy did not pass it on to any of his children, and the girl passed it on to all of her children. So that one's pretty straightforward. There's 3 that are pretty important. This, we talked about buzzwords. This is a big one. So if they talk about a disorder where somebody has this and they mention ragged red fibers, they did for us, like people told us that they were going to, and they did. If you see ragged red fibers in the question, you can go ahead and assume it's going to be this myoclonic dystrophy with ragged fibers, the MRF, and that's going to be mitochondrial inheritance. And then this Leber hereditary optic neuropathy is important. And this one also too, I'm not going to say all that, but that one's important as well. So make sure y'all know the characteristics of those, the mutations, the genes, and all that. Okay. Yeah, I've got a couple more, and then I'm gonna give it over to Sarah. So the locus heterogeneity and the allelic heterogeneity, those are ones that trip a lot of people up, and they messed me up. I don't know why. They shouldn't be difficult. And I think Dr. Sobering told us this too. They shouldn't be difficult because it's different genes versus the same gene. But for some reason, every time we saw this in an exam, it tripped me up and I had to think about it. So make sure you know these pretty well. I don't even have a great like mnemonic or way to know them. If anybody does, feel free to throw it in the chat. But so locus heterogeneity is going to be the opportunity for mutations at different loci or on different genes. They're going to be completely different genes with different mutations, but that cause the same disease. So osteogenesis imperfecta is an example of this because there's 2 different chromosomes that can have mutations that are going to lead to the same disorder. So chromosome 17 and chromosome 7, they're not even on the same chromosome, but they're going to cause the same problem. You can have either one of them and have the problem. Same thing with the ones down here. Charcot-Marie-Tooth, you guys will talk about SCID, the retinitis pigmentosa. Yes, that's going to be locus heterogeneity. So 2 different, completely different genes, different chromosomes, everything like that. Now, allelic heterogeneity on the next one is going to be different kinds of mutations, but on the same gene. So it will still be different mutation, different kinds that can cause the same phenotype, but it's going to be on the same gene. So for example, yes, neurofibromatosis, there's thousands of different mutations on that gene because it's a mutation hotspot, and they're all going to cause the same disorder, and it's on the same gene, same chromosome, but there's different areas that it can be mutated. There's different types of mutations that can occur, and they will cause the same problem. Just note that it's on the same gene for allelic. And this also has something to do with cystic fibrosis. So yeah, you can have a couple of different mutations that will cause cystic fibrosis, and you guys will talk about cystic fibrosis for like the rest of your life. But the deletion of the phenylalanine at the 508 is the most common one, but then there's, like it says here, thousands of others, frame shift, missense, all of that. So I know there's an example with compound heterozygosity and cystic fibrosis, and I didn't include it on here, but that's pretty important as well, where you can have the different types of mutations like that. Like one parent can be a carrier of one type of mutation, and another parent can be a carrier of a different type of mutation. And if the child inherits one of each of those, they can be affected, and they would be a compound heterozygote. I probably should have included that, so sorry about that, but definitely know that. Okay, this one, I will tell you specifically, I remember there being an exam question on this, and I remember it because I think I got it wrong. So there was like some discrepancy amongst the students of like what the correct answer was, but it's kind of hard to tell sometimes the difference between incomplete penetrance and variable expression, because they'll give you like a whole situation about a person and their family, and they like so-and-so has these symptoms and so-and-so has these symptoms, and they might be similar to one another, but they might be like in different severity. And so it's kind of hard to determine what you're looking at here. So incomplete penetrance is going to be something that disrupts an autosomal dominant disorder. So you'll look at the pedigree and it looks like it should be autosomal dominant, but then there's like one person that looks like they might be messing it up. Like it might look like it's recessive on that left side because it skipped a generation, but then it's like it doesn't skip a generation on the right side there, so it looks like it's dominant. It is tricky because it's hard to tell, and they would have to give you enough of a pedigree for you to be able to determine that like one of the sides is dominant and there's enough information there. But it's important to know this one and the following ones just because it's kind of easy to get them mixed up, especially the way that the exam questions are going to be paragraphs about, you know, people and their symptoms and stuff. It's not going to be super straightforward. So make sure that you know the difference between this one and then the next one, the next slide, Kishore. Oh, I have another slide on this actually. So this is basically just reiterating that. This is from y'all's slides as well. Sometimes it can be dependent on age, but that's also different from anticipation, which is another important one. So there's a few of these that kind of go hand in hand. And the age dependent thing, Huntington's, yes, they like drilled this into us. Huntington's has a high penetrance, but it doesn't have an early age of onset. So earlier when I was talking about diseases that have to be obvious when a child's born, that was when the caveat was, but that was recessive. But with this, Huntington's disease, for example, has like a way later age of onset. So people might develop it as they get older. And that with that specific one, that'll usually be a tip in the question. They'll say like, so-and-so's dad, this, and like they got older and things like that. But breast cancer is another example of age-dependent penetrance because 95% penetrance in familial breast cancer by the age of 80. So you're not going to have it when you're younger, most likely, but it's age dependent. So that goes with this incomplete penetrance. And then this, so like variable expression, it's similar. I know specifically the ones that they gave us were that xeroderma pigmentosum, because it has to do with being in the sun and having exposure to the sun causes like way more severe damage with the UV radiation than it would for a normal person. But then, they talk about there could be other variables involved. Like if you have a child that has it, you might be more prone to keep your second child out of the sun, things like that. But the variable expression is basically just what it sounds like. If two siblings are affected with the same disorder and they exhibit different symptoms or a different severity of symptoms, that's going to be your variable expression. Okay, and then germline mosaicism. Kishore threw this slide in here, and I definitely was not prepared to talk about it, but I can just, you want to tell us one, Kishore? I didn't even look at this.
Yep. No worries. So no problem. A germline mosaicism, right? So let's just look at a case first. So a healthy man has 2 children affected with osteogenesis imperfecta with different parents, right? The father has another child but except he is normal. So what is really happening here is because of a germline mosaicism. Remember, in a germline, what is a germline, first of all? Let's establish that. Feel free to unmute yourself or put in the chat what germline mosaicism is.
Gametes.
Gametes, exactly. And what do we know about mosaicism, right? Are mosaicism like the exact same patterns all over the place, or is it different patterning?
Different patterns. So it'll be like mutations in the gametes, the ovum and the sperm.
Exactly.
And then the parent might not have it, but the child, when they have children, they will definitely show it later on.
Exactly. So think about it this way, right? Let's say this man, right? Clearly he has a genetic mutation that's affecting his germline cells, which is his sperm, essentially. But what's happening is that some of his sperms are good, right? They have no mutation. They don't have the mutation that is causing OI, osteogenesis imperfecta, whereas some of his sperms that were made later on, they do. So it shows you that you can have, within the exact same person, you can have germline mosaicism based off of the nature of the mutation and how the whole spermatogenesis process is. Okay, that's just something to keep in mind. Typically, they don't like to test germline mosaicism unless you have very keen and specific like tests and examples to kind of show it, right? They have to give you something along the lines of like, oh, half of his sperm has viability, whereas the others seem to have this particular thing. And then you can be like, oh, okay, germline mosaicism. So that's just something to keep in mind. A lot of the ones that Cassidy already mentioned, those tend to creep up more because those are things they can really test your genetic understanding of, because germline mosaicism is hard to do so without giving you a lot of background. And if they give you that much background, then you likely could jump to the answer right away.
Yeah, thank you. I think I want to say there was like one specific disorder that they gave us that was like really consistent with germline mosaicism. So if you see that, if they give you an example, just know that. But otherwise, I would say, like Kishore said, it's not like super high yield.
Do you want me to take this one?
I'll finish this one. And I think Sarah's gonna go, but this one's super important. This one will come up a lot just because it's Huntington and it's a triplet repeat. I remember we probably got tested on this like multiple exams, but know for sure that anticipation means that as the generations go on, the symptoms start to onset earlier, basically. So they usually would give you an example of like a man, and they would say he's becoming forgetful and blah, blah, blah. And he's having all these muscle twitches or something. I don't even know. I think that's right. And then his father had a disorder and these symptoms onset when he was in his 60s. Or they might say, his grandfather had this disorder and onset when he was in his 80s, and his father had this disorder and it onset when he was in his, this. And then this man might be 30 something years old. And so that is anticipation. And it has to do, this is really common with any of the triplet repeat disorders, but Huntington is the biggest one that they like to teach here. But it has to do with the polyglutamine tract. And then if there's more, it's unstable. The triplet repeats are unstable. So there's this thing called slipping. And as the repeat size gets larger, then the disease severity gets earlier and earlier. And that is anticipation. And that I can like almost guarantee you'll probably get asked about that because it was super important. Okay, Sarah is gonna take this imprinting from here because I don't even understand this to this day. So I'm gonna listen to her and learn with you guys.
Thank you. For the question in the chat, so anticipation and incomplete penetrance, the difference is with anticipation, it's going to show up at some point. It's just that it'll be age related or it's related to more factors than just the inheritance. Where incomplete penetrance, that person's not going to show it. Like they're affected, but they're not showing anything. So you don't know they're affected unless someone tells you that they're affected. Anticipation will show up. And that will be for the triplet repeats, yes. So all the triplet repeats, you should just automatically in the back of your head associate that with anticipation, and that should be enough for you to figure out the answers on the exam. Okay, let's talk about imprinting. So with imprinting, if we're looking at, sorry, let me just see if I can annotate so that I can do this. Okay. So when we're talking about these specific genes, we're talking about chromosome 15. You have your SNRPN and your UBE3A. Normally, you have methylation on your UBE3A from your paternal chromosome 15 copy. And you have methylation at your SNRPN on your maternal chromosome 15 copy. So methylation means silencing. So the parent that has the methylated copy, that gene is not expressed from that parent's copy. So when it comes to imprinting, this is talking about how a disease can manifest if something happens to, let's say, this paternal copy of SNRPN. If there's a deletion of it or a mutation causing it to be inactive, then your mom's copy is already methylated. So that's not going to work. Now the paternal copy is also out of service. So that's where you would start to show the symptoms. And it's the same thing for the UBE3A. So depending on the parent's copy that is expressing that gene, then that's what, if something happens to that functional copy, then that's where you would see that disease manifesting. I think we can, is there another slide on this? Yes. Okay. So that's where these come in, Prader-Willi and Angelman's. So with Prader-Willi, your maternal gene, sorry, no, the paternal gene is the one that's deleted. So if the paternal gene was the only one that was functional because the maternal is imprinted, is methylated, sorry, then this is where you would see Prader-Willi manifesting. If something happens to your only functional copy, then you have nothing else. So Prader-Willi will be the paternal allele being deleted or mutated. Angelman will be the maternal UBE3A being deleted or imprinted. I hope that makes sense.
And for these, they can be very buzzwordy too. So first aid is a great example of where you can kind of incorporate these kind of practices in. Seizures, ataxia. If you hear laughter, then you can be like, boom, it's Angelman, right? So those are a couple of ways you can kind of navigate this, even if you don't have the strongest like genetics background on this. Just be aware of what Sarah said. Remember which one is mutated, which one is silenced, because they can test you. They might just give you all the symptoms and they can say which one's methylated, which one is mutated, etc.
Okay.
Wait, just to confirm, when you said that they could ask which one's methylated, which one's mutated, so when it's methylated, that's the one that's being imprinted. So for instance, if the UBE3A was methylated, therefore imprinted, that would be Angelman in that case?
So the methylation part is naturally what happens. So a lot of people will associate methylation with, oh, this is going to cause Prader-Willi or Angelman. It's not. The methylation is already there, even if you don't have Prader-Willi or Angelman. All of us have 2 copies of our chromosome 15. On our paternal copy, we have methylation at the UBE3A. So when we're talking about Prader-Willi and Angelman, we're talking about the mutation or the deletion of your other copy, the functional one. So think of imprinting as one thing, and then the mutation will be on the other one.
Okay, and the one that has the mutation or the silencing is the syndrome at hand?
Yes.
So, okay, perfect. I got it then. Thanks.
Okay, No problem. I know this is confusing. For some reason, they confuse everyone every single term. So I hope that made sense to you guys. I think we can go to the next slide and someone else can take over because no.
Okay, I'll do the definitions. Since I made you guys talk so much with all these slides. The key things with these is to know very much the terminology linked with the pathology, right? It's easier to memorize it that way than having to sit down and like do Anki cards for the next, 10 years. So let's look at the dominant negative mutations. So what it does is it exerts dominant effect. A heterozygote produces a non-functional altered protein that also prevents a normal gene product from functioning, okay? What that means is you created a copy of something. That copy is then going to go and mess up the normal versions of that exact same copy, right? A great example of this they've given here is P53, but I can tell you right now, you're going to see OI, osteogenesis imperfecta, in terms of the dominant negative mutation of one version being created that affects the other, which is the mutated copy affects the other. So my advice for you when going through this kind of key terminology definition kind of process, link the diseases with them. Okay, the linkage disequilibrium, you really don't need to know. That one is just pulled in from first aid for just understandment purposes of further things, but it's not needed at this point. Mosaicism is going to be important because somatic mosaicism is very much different from gonadal mosaicism. Can someone, since we're all talking so much and I want to make sure this is interactive, somatic mosaicism, what is that going to present us?
I think of it as like you have different color spots on your skin. Like let's say someone has like white and black dark spots all over their body, that will be like somatic. You can see it on the body. And the other one is hidden, it's in your gametes.
Exactly. So a person with a somatic mosaicism, actually a fun fact that comes back from term one is calico cats. If you guys remember calico cats, they have red, black, and white spots, right? And that is somatic. Soma meaning body, so it affects the person's somatic cells that are not germline. Of course, the gonadal or germline one, they can call it different things, is affecting specifically the gametes, the products of which you're going to have genetic, continuation of your genetic line, right? So those folks likely won't see any, phenotypic presentations of a pathology. What they will likely see is the example that you guys saw before, where the dad had a gonadal or a germline mosaicism that basically didn't allow for, or you saw that he got married twice. One, he had only kids that had the OI problem, and then out of the blue, the kids didn't have an OI problem. Now, follow up on that. What is osteogenesis imperfecta? What is the inheritance pattern for it?
Dominant negative is more severe.
Dominant negative is more severe, but autosomal dominant was what I was looking for. But exactly, it's important to remember these. Make sure you test yourself on that, right? Like, let's say you come across a pathology, associate it with what you know from it so that you can constantly refresh your knowledge on it, because some of this stuff isn't intuitive, it's just based off of repetition. It's going to make sense. Let's move on. Locus heterogeneity, I think some of these things we covered with you guys. Mutation at a different loci or a different gene altogether, right? We're talking about like different chromosome, different part of your genetic composition. One can be on chromosome 7, the other one could be on 17. Great example, cystic fibrosis, like Cassidy said. Allelic heterogeneity is a completely different thing, right? Different mutation on the same loci. So same loci, that meaning we've identified in that location as being problematic. In that, you can have at different points various mutations. You can have like, okay, on the 5th phenylalanine in this position, you have like a likelihood of you getting like beta thalassemia or something like that. So just remember to go back and see which one is allelic and which one's hetero. My practice for this one was literally take a white sheet of paper, write down each of the specific pathologies based off of these broad categories, and it will make sense. I swear to you, that's how I got through it. Okay. Heteroplasmy, presence of both natural and mutated DNA, resulting in variable expressions of the mitochondrial inherited diseases. You saw this already. Very simple. The amount of mitochondria that the mother has given to the offspring is going to be dependent on how much of the pathology is presented, or the phenotypic presentation is going to be. The uniparental disomy, this one, we didn't get tested on it so much. Just be aware of that. Offsprings often receive 2 copies of the chromosome from one parent, no copy from the other parent. Heteroidosomy, iosomy indicates a meiosis 1 error. And then, actually, Sarah, did we get tested on this that much? I think all we had was Prader-Willi and Angelman. I don't think we had any germline issues with this.
I think we might have, because isn't there like a way that you can, yeah, you can,
When we talked about,
you can get Prader-Willi, yeah.
I think with uniparental disomy, yeah, I think we did talk about how if you're only born with one functional copy and it's the copy that's methylated, then you could manifest that disorder. So if they say that there's an issue with the parental or the paternal copy of chromosome 15, and that's the one that you're looking at as the functional, then you already know you don't have any functionals.
Just be aware, I think Dr. May and Dr. Martin might have taught this section, so just be aware they tend to put in a couple of questions on Prader-Willi and some on the germline issues too, so just keep that in the back. Codominance, variable expressivity, incomplete penetrance, all sound like they're the same thing, but they are not. You have to keep that straight. So codominance, both alleles contribute to the phenotype of the heterozygote. So for example, your blood type, right? Like you could be A, you could be B, you could be AB, and then like there's pathologies too with the alpha-1 antitrypsin deficiencies and the HeLa groups and et cetera. So variable expressivity, can't pronounce anything. Patients with the same genotype have various phenotypes, right? A great example of that is 2 siblings who have neurofibromatosis. One has hardly anything, the other one has huge tumors, benign tumors, right? And remember, it is a tumor suppressor, so you will likely, they can ask you a follow-up question by saying like, yes, we know that they have NF. What is something else you can get is potential cancer, because NF or neurofibromatosis gene 1 is a tumor suppressor one. Incomplete penetrance, it's not all individuals with the same genotype will show the mutant phenotype. Example in this case is also NF. I don't know if you guys covered your BRCA1s, but I will say that will be tied in later. But example, a mom has the BRCA1 gene mutation. She doesn't present with any chances. Well, she doesn't get cancer for ever, and then she passes away of natural causes. Her daughter, on the other hand, gets it, no idea why, but remember, incomplete penetrance can be a way to describe the fact that you don't have at every single point, you don't have a affected individual or every single line of the generation. Pleiotropy, one gene contributes to multiple phenotype. Great example is PKU. We personally covered it this in term 2. And it can present with just one gene, right? One mutation can literally have downstream impacts on everything from brain development to with, skin composition and et cetera. So anticipation, you guys know, Huntington's is a great example of that. It really depends on when you've seen the onset, early or late, just be aware. And then loss of heterozygosity. This is basically a double hit theory, okay? I don't know if you guys have covered this, Lynch syndrome, and then Li-Fraumeni syndrome. Both of them follow, I think they follow tumor suppressor genes, and both of them get hit either one during the genetic composition or the two being the environment, or both of them getting picked out during when you're doing, well, genetics or passing on of your germline. Okay.
Didn't do that.
Yeah, that's an FTM2 thing, I think. So ignore that last one until you guys get to that.
Correct.
Okay.
Sarah, you want to take it away?
Yes. So you guys do have this flow chart in your lecture slides. I remade it because it's the ugliest flow chart I've ever seen in my life. So if you memorize this flow chart and follow the questions in order, you will always come up with the right answer for your pedigrees. So you're always going to start with, are multiple generations affected? When we're talking about multiple generations, we mean, is it a vertical inheritance? Are parents affected and passing it on to their children who are becoming affected? That would make it dominant. If it's not, it's recessive if you see horizontal inheritance. When you get to your dominant side, you're going to say, okay, is there male to male transmission? If you see a pattern of only mothers passing it on to their sons, or only fathers passing it on to their daughters, if you don't see fathers passing it on to their sons just as equally as their daughters, then you would talk about this being X dominant. So if you follow these questions, you should be able to come up with the correct answer for every single pedigree. Just memorize this flowchart. I promise you, it'll always be right. The only things you have to consider are, like we were talking about, incomplete penetrance. If you see one person that is messing up your whole pedigree, try to ignore that person and work that in with one of the terminologies. Incomplete penetrance. I think that was like the main one that would kind of mess this up. So I think we can move on to the next slide. And this is a clicker question. But again, yes, so definitely memorize that flowchart. I don't think I spent more than 20 seconds on any of the pedigree questions on the exam, and I didn't miss any of them, so it works.
I just want to hone in on the point that these are the type of questions that you're going to see, except they're not going to give you this nice short vignette. Likely they'll give you something along the terms of, here's some, pathologies, here's some, co-symptoms of things. So it's not, they're not always going to tell you sickle cell disease. They might just play around with it and just give you that too. Remember, this is how they kind of build your kind of first order, second order capacity. Quick answers, guys, quick answers. You guys have 30 seconds. Giving you guys a little extra time because I know this question. Okay, let's see how you guys did. Let me stop the timer. And majority of you, we had a little bit of a distribution. Majority of you eventually came to the right answer. And that is B, okay?
Yeah.
So who are your,
oh, sorry, go ahead, Kishore.
Go for it, explain away.
So when you're looking at this, if you're talking about sickle cell, then you're talking about what type of disorder? What's the inheritance pattern?
Autosomal recessive.
Autosomal recessive, exactly. So any of these that are filled in, you already assume that they have two copies. They have two affected copies. So for mom number one up at the top here, she's affected. So if you're getting one copy from mom, one copy from dad, all of her kids are going to be obligate carriers, right? Because they all have to get at least one copy from her. So row 2-1, 2-2, and 2-3 have to be obligate carriers. And then you move down to the bottom and you see that these two children are affected. in order to be affected, if none of the parents are affected, both of them have to be obligate carriers because you get 1 copy from mom, 1 copy from dad. So that would also make 3-1 and 3-2 obligate carriers. So that's kind of like the train of thought that you want to have when you're trying to answer these questions. Does that make sense?
And just a pro tip, if you guys are daunted by this whole process, I highly suggest you use the flag feature in the exam, save them, come back so that if you have extra time, you can really give it the due diligence it needs because they can ask you questions like this on the exam. Okay.
And
Let's
you
do
also
another one.
highlighter. So like if you want to like highlight some of them, if you think like, oh, this person's going to be a carrier, this person's going to be a carrier, like while you're thinking about it, you also can do that as well. I've done that.
So while you guys are doing this question, we got some questions in the chat. So why can't just mom or dad be a carrier? If you have affected children and it's autosomal recessive, you need 2 copies in order to be affected. And 2 of those children were affected in the 4th generation. So that's how you know that each parent has to have at least 1 mutated copy of that gene. No problem.
30 seconds, guys. Quick answers, please.
Oh, real quick while we're doing this still. So the reason that the first dad was not affected or was not a carrier is because none of the children were affected. They were all carriers, so they only needed 1 copy. And you know, mom has 2 bad copies, so they're all getting 1 from her. If dad was also a carrier, then there would be a chance that one of those children in the 2nd generation was affected because they would be getting 1 mutated copy from mom. And then if dad is a carrier, they could possibly get that other mutated copy.
You see how Sarah's going through this, right? Like that mental process of assessing each of the likely candidates. Do that with each of Dr. Sobering's question. Because remember, you can always land on the correct answer, right? It's not the concern here. It's about making sure the variations that they can ask of you, that you're also capable of answering those too. Okay, I'm gonna stop talking and give you guys what you guys clicked in. Let's see how you guys did. Awesome. Okay. The answer is C. This is the one that Cassidy went over and Sarah went over, and I also went over. It's Prater-Willi Angelman. Remember to associate each of the pathologies with the term and be comfortable with understanding what the definition of each term is. Okay. Huntington's, anticipation. Heteroplasmy, MRF. Okay, we talked about that in terms of mitochondrial imprinting. Prader-Willi Angelman, we talked about that. We talked about the methylation, etc. Locus heterogeneity, osteogenesis imperfecta, mosaicism, also a great example of osteogenesis imperfecta. Okay, awesome. Let's get this out of the way. Let's do this.
Okay, let's talk about PCR. So with all of your molecular diagnostics testing, associate them with the disorders, please, because they get kind of crazy. You don't have that much time to be thinking about all the possible tests to do a specific disorder to figure out if someone has a specific disorder. So with your triplet repeats, PCR, always think PCR. It's 99% of the chance going to be your answer. So the way that PCR works, I think everyone kind of has that down already, but you're pretty much just amplifying that DNA and you're giving yourself a cDNA copy, which is like a reverse transcript of the DNA that you originally put in. So when you see Huntington's, myotonic dystrophy, Fragile X, you're always going to pick PCR. That was the answer. With PCR, it's going to be PCR gel electrophoresis most of the time, because I think Cassidy had a slide that was earlier on that was showing a gel. And when you look at the anticipation, the more copies of that triplet repeat that you have in your DNA, the slower that band is going to travel on the gel. So your gel electrophoresis would be your way of seeing what your PCR is producing. Okay, so for your karyotyping, it's going to be mainly associated with your trisomies because you're getting a whole extra copy of some chromosome. So one thing with the karyotyping is you're always looking for really big mutations because all you're looking at here is the banding patterns. So it has to be greater than or equal to 5 megabases in order for it to show up on G-band karyotyping. So if they give you, let's say it's trisomy 21, they describe a patient that has Down syndrome and they're asking you what method would we use to confirm this, then you would say your G-band karyotyping. Next slide. RIFLIP. So with your RIFLIP, you're looking at restriction fragments. So it's going to be pretty much the same idea where you're doing PCR, you are getting some, sorry, distracted by the chat. So you're getting some copy of this, these splice sites. So depending on if you're using EcoRV or BAM-H1, then that's going to give you different types of splices. I think on the exam, they probably just asked us a question about if you would get like a blunt end or a sticky end. And a sticky end just means that they don't line up. A blunt end would mean that they do line up, like in the EcoRV. And then you could use this for sickle cell. And for sickle cell, you can also, I believe, use your ASO, but just be sure to associate them with both. And then maple syrup urine disease would also be one that you want to associate with your RIFLIP. Next. Okay, ASO. ASO associate with your cystic fibrosis and your sickle cell, but mainly your cystic fibrosis. So you're talking about very small mutations, and they have to be small enough where you can sequence them out and say, I know this mutation is like Delta 508, that one for cystic fibrosis. So if it hybridizes to that spot where you have the mutated sequence, then that's telling you that this person does carry that mutation, but you have to know the specific mutation in order to create that blotting point. Next one. cDNA is usually the one where they talk about cancer. Cancer is used for this because with this one, you're looking at expression. So if you see a gene that is insanely overexpressed, then you can say that's probably cancer. And the way that you would look at it is your cDNA microarray. So you would see red as overexpression, a bunch of red spots on your chip. And then with underexpression, if for some reason a gene is not working or it's being silenced by something, then you would get green. Okay. And then SNP microarrays, those are going to be your single nucleotide polymorphisms. So just know that that's pretty much all we needed to know for it, is that it's a single nucleotide that's being changed. Array CGH, when you look at this diagram, you're either going to have things coming up on your right-hand side or your left-hand side, and it's going to tell you something is either overexpressed or underexpressed. So you can use these for your triplet repeat expansions, but with your triplets, it's going to be more PCR. Like when they ask you on the exam, they're usually going to associate it with PCR. If they do show you this and they're telling you that there is like overexpression of CAG, then you automatically know that they're talking about your Huntington's. So you can use that for triplet repeats. You can use it for balanced translocations. And so translocations means that like you would expect to see it down here where this red circle is, and instead you end up seeing it like here. So it's still there. You're not losing or gaining any information. You're just moving it around. And then you can also use this for trisomies and for your APC. Next one. Fish and skyfish. This one I hate because it's terrible. But with your rapid fish, the one key term that you need to know is that it's going to take longer because you need it to be in interphase. So everything that you are using rapid fish for, you have to arrest those cells in interphase. So that one's not going to be as readily available. For your skyfish, that's going to be painting each chromosome a different color. So that one would also be good for your translocations, because if I expect to see, for example, if I expect to see both of these as purple and for some reason half of it is red, then you know that something from over here got translocated over there. So it's pretty much looking for what colors are not matching up.
But isn't rapid fish like really fast? You do it on infants in the start, so you only need interphase, but for the skyfish, you need to freeze them in metaphase.
Oh, I'm sorry. You're totally right. Yes, I'm sorry. I got mixed up with that one. But yes, metaphase is the one that takes longer. Rapid fish, if you think about your cell cycle, most of the time you are in your interphase. So that one, you're right, is going to be the rapid one. When you are waiting for metaphase, that one is going to take longer. Okay, we can move on. Next gen sequencing, we didn't really get tested on as much, but if they talk about how you have no idea what's going on and you need to figure out what type of mutation this person has or disorder, this is where you're pretty much sequencing their entire genome to figure it out.
So we had a joke in our term was that whenever you see next gen sequencing, it's probably the answer, okay? Because it's so advanced in terms of the other testing methods. Like most of the other ones have been around for a while. So if you see it, don't automatically pick it, but most of the time, read the stem, really make a good educated guess. If you're still lost, it's probably next gen sequencing. Okay?
Yeah.
Unless it's like an obvious one, like, DGeorge, fish and chips and all that stuff, okay?
Can I ask a quick question about the different processes and stuff?
Mm-hmm.
Do we have to know like the specific steps or just like a general overview of what we're going to get when we do the tests?
I think it's more of a general understanding of what you're using and like what you have to know to do the test and what your outcome is going to tell you. So if you're talking about like your ASO, I have to know the specific mutation and I'm going to get either a yes or a no. But when you're talking about your PCR, you're going to get, if you run a gel after it, let's say you're talking about your Huntington's, depending on how many copies of that triplet repeat you have, then your band is going to move slower. So different tests are giving you different amounts of information. So that's the thing that you should be familiar with, because based on what they ask you on the exam, you can figure out what test is able to come up with the answer I'm looking for.
Okay, that makes sense. Thank you.
No problem. This is a really good summary table. I used this one. I would definitely look at it, especially if you don't have much time to go over these. Just kind of look at these. It tells you which ones are usually going to be associated with which test and what the criteria are. So if it needs to be greater than 5 megabase pairs, if it needs to, if you need to know the mutation or don't know the mutation. Next slide.
You do need to know this though. Please look at it. We have in our drive, we have a kind of a condensed version of this too, with a little bit more details with other, because like we've given you some pathologies here, but like there are other ones associated with different diseases. So honestly, like Sarah said, if you don't have time to memorize all of them, take a quick look right before the exam. Really like just memorize, associate based off of buzzwords. Sorry, getting tired. And you'll be okay. Okay, just make sure you do that. It is heavily tested. You need to know it.
In the chat, for the PCR and array one, if they're giving you array like for a triplet repeat, it's going to be obvious. So you know already the code, the code that's going to be repeated. So if they tell you they're talking about Huntington's, like they're talking about Huntington's, they're not trying to trick you. But if they ask you what is the best test for this disorder, the triplet repeats are PCR. And then here's a question.
And you will have to read some of these, right? Techniques, right? It's not just straight up like you need to memorize like every single like factor or thing that you need to do, like an ASO, snip chip, or any of those things. Just be aware that they do have questions on your quizzes and in the exam that just straight up ask you how to read these things. It did take some of us a while, including myself, but more practice is the only way to kind of get over that little hurdle of like, what am I looking at and how do I interpret it?
I still don't know. I'm not gonna lie.
It's okay. We're done with FTM 1 thing. Good.
Make Sarah like run through these like before each exam last term. I'd be like, okay, tell me about those things again real quick, like in the morning, because I didn't learn them. So I would recommend learning them the first time and not being like me.
It helps.
It does. But you guys get through term 1. You don't have to look at it until BSE. So that's just the boon of making through term 1.
Yes, Lexi gave very good advice. Prater Willie likes fish and chips. And did George just likes fish, right?
Mm-hmm.
I think it was something like that. But it was like the little mnemonics and little tricks like that that we honestly used for a lot of that.
But a question what CHIP stands for.
Do you recall how many questions there were again on the exam? I've been hearing different numbers. And that they had like 3
in total questions?
Yeah, for the exam 1. Mm-hmm.
We'll have to look, but the first one is shorter. The first exam is shorter. It was shorter for us as well. I think it's like maybe 40 per section on the first one, but then it'll be like 46 or 48 per section on the subsequent ones, but you'll get more time. So I think you guys will get an email this week that breaks down the blocks and the times and all of that. And then some of those questions will also be experimental.
Okay, I'm gonna close the poll. Let's see how you guys did. All right. Hopefully you guys aren't looking ahead at the answers, but the answer is C. Get this out of the way.
Sorry, that was me. I didn't mean to do that.
You're good, you're good. Now, let's break this down a little bit. So why is it C? Okay, first you have to understand that what you're looking at right now is normal ASO. That's right here, okay? Normal ASO. Then you have the pathological one, right? That you're assessing for. So, and when you're going through this, then you have to compare, right? The normal ASO this person has, so right there, right? They have the normal and they have the abnormal. So what does that mean, guys? What is the genetic breakdown of this person?
Heterozygous.
Heterozygous, right? If you can break it down by these kind of simple ways, then you have a better understanding, right? I don't want to spend too much time because we have so many things to cover with you guys, but you guys can see heterozygous, this person has only the mutation copy, so they're homozygous recessive. And then here, this person has only the normal copy, so they're homozygous dominant, and they're A-OK. And here, this person is once again heterozygous and likely a carrier, okay? They can ask you questions as simple as this. So the questions that we're giving you are representative, just the question stems might be altered a bit, might be made longer. So just be aware of that. Okay, next one. There's a lot of answer choices. Okay, go for it, guys. But feel free to unmute and ask you guys any questions.
How large of a deletion? Is it over 5 million?
I would say yes. I would say it's over 5. Yeah. I should have specified more.
I do remember that being a cutoff. I forgot about that.
Yeah. Okay. Let me call it. See how you guys did. So, the answer is A. Why? Because this is particularly talking about what? Microarray CGH, right? So microarray CGH, let's get this out of the way, is a query that tests the entire, genome in one test, right? It offers increased resolution compared to G banding. Obviously, it does not test for triplet repeat expansions or SSRs, okay? Too small. Balanced translocations, discuss more in cytogenetics, and then SNPs are very small. So just be aware, they will associate a pathology. They won't ask you like how I did here. Oops. They won't ask you like how I did here, which just like a general stem. They're going to give you a pathology. So I can see why you guys would be confused on this one.
With that question though, I don't know who asked, but the fact that he didn't tell you it was a 5 megabase one is how you would say like, I'm not 100% sure I'm able to choose G banding. So that's why microarray CGH would have been the better answer for this question, because you don't know how large, like you don't know how large is large. So that's why you would pick microarray CGH. It's going to give you more resolution compared to your G banding.
Oops. Why can I not pull? Sarah, I need technical support.
What's up? It's there.
Is it polling?
Oh, I don't know.
Oh, okay.
This is what happens during lecture.
Okay.
Yeah, it's not.
Okay, well, we can just go over this one and hope the next one works.
Okay, so this is PCR. So mouse is all sorts of weird today. Okay, so you do need to know with your, once again, like Sarah said, triplet repeat sequence or tests and sorry, your triplet repeat disorders, you do need to know that they will go with PCR, right? How would you know if you're going to have an earlier onset versus later onset?
Number of repeats?
Number of repeats, AKA the weight of it, is basically being assessed here. So if it has 100 repeats, then you're looking at someone who's going to have a later or earlier onset.
Later on.
Later or early, sorry. It's early, and if it's much more like they have fewer expansions, right, or repeats, then it's going to be later. Okay.
Okay, so with your receptors, I don't know how far into this you guys got, but your exam's this week, so we're not going to be having another review. So we'll go through it anyway. Your steroid receptors, we know that they're going to diffuse across the cell membrane, and that's going to have its receptor in the nucleus. So you're just looking for the buzzwords nuclear receptor. You're going to look for cholesterol because most of the steroid hormones are made of cholesterol. But if they talk about a hormone that is just changing gene expression, either up or down regulating it, then it's probably going to be your steroid receptor. For your gated ion channels, those are going to be mainly talking about either ligand gated or ion gated channels. With your ligand gated, you need some sort of ligand to come and bind to the receptor. Sorry, I can totally annotate to make this easy for you guys. Okay, so you're looking at, this is your ligand. It's going to bind to its nicotinic receptor over here. And that's going to cause the opening of this ion channel, and that's going to cause the influx or efflux of some ion, and it's going to cause something else to happen downstream. So that's what you guys want to look for with your ligand-gated ion channels. Receptor tyrosine kinases, know that that is associated with your insulin. And then you have your dimers that are going to hybridize, and that's going to make your receptor. So you have two subunits that come in, connect to each other, and then that hormone or insulin, whatever you're talking about, is going to combine here, and it's going to cause phosphorylations and a whole bunch of downstream effects. Next one. Okay, these you're going to get a lot more in depth on when you get into autonomics. But one thing that you should kind of understand now is the different pathways for the G protein coupled receptors, because they will make your life a lot easier when you get into autonomics. So with your GSs and your GIs, you're talking about cyclic AMP. That is your second messenger. Always associate that with it. GI stands for G inhibitory. GS stands for G stimulatory. So you're either upregulating the cyclic AMP or downregulating it, depending on which type it is. And so cyclic AMP is going to cause PKA, or protein kinase A, to do its thing, and then you're going to have downstream effects. With your GQ, that's going to be your phospholipase C, and it's going to be cleaved, it's going to cleave your PIP2 into IP3 and DAG. So your second messengers for your GQs are your IP3, your DAG, and calcium, I think, is also considered one of the second messengers for your GQ. So know that that's going to cause something to happen with calcium. You're upregulating the calcium. With your nitric oxide, I don't know if you guys went over this one yet, but if you did, then you know that nitric oxide is going to diffuse across the cell into the skeletal muscle cell. And usually we're talking about blood vessels when we're talking about this. So this is going to be a smooth muscle cell, and it's going to cause your cyclic GMP and your protein kinase G to do their thing, and then you get a decrease of calcium. So your second messengers are cyclic GMP, IP3, DAG, and calcium. I don't know if DAG is considered one, actually. And the other one is going to be your cyclic AMP.
Were you asking if DAG was a second messenger?
I'm not 100% sure.
It is.
It is, right?
Yeah,
Okay.
yeah.
Good. Because sometimes they tell us, like, just look at calcium and IP3. Sometimes they do include DAG. So just know that all three of those are going to be somewhere in there. And then this is just kind of a summary slide. You're going to get a lot more in depth when you guys start talking about your autonomics, but this is kind of as far as you need to know, and you guys can use this while you're studying for it. Next slide. Clicker question.
You guys are going to cover this week enzyme kinetics and all of that. So this is a little ahead for you guys. Bear with us. We're trying to introduce it to you guys now so that you don't get overwhelmed the several days before the exam. So that way you guys can think of this as your pre-read process.
Yeah, feel free to like completely tune us out right now, but we are just trying to give you guys some exposure, because we're not going to get a chance to do any reviews with you guys before your exam, because it's this week. So this is, it was talking about your second messenger lecture. I believe you guys are doing that tomorrow, not 100% sure.
Make sure you guys click in. Your attendance depends on it. Not part of your 80%, but just as a fun 80% here. So
did you guys cover enzymes yet? Anyone?
Yes.
Okay, good. Just making sure.
okay, let's look at the answer. So most of you guys went for D, and that is the correct answer. Okay, it leads to a decrease in enzyme affinity for the substrate. Now, what is this called?
KM.
AM, sorry?
KM, the affinity for the substrate.
Exactly, KM. I love it. Awesome. Exactly. So this is going to come back again and again, but in different capacities. You're going to talk about it in CPR. You're going to talk about it in blood. You're going to be talking about it all over the place. So it's important that you guys learn the fundamentals of it before we start jumping into other ones. Okay. feed-forward activators, the small sigmoid curve is shifted to the left, resulting in a smaller K. Okay. And then the effector of the feedback inhibitor, the sigmoid curve shifts to the right, which results in a larger K. Okay. Let's do this one, something based off of what Sarah said. And we need 4 answer choices. Go for it. Now I have a timer, guys. I don't know why it wasn't working before, so.
You're welcome. I totally understand. This is exhausting. It's very long for you guys. Thank you guys for sticking around. We're just trying to get as much information to you guys as possible before your exam.
Yep.
Thank you guys for doing this for us.
I know, for real.
We never realized how hard it would be to remember FTM1, but it's really difficult. But it's
I
helped
swear.
us, so thank you guys.
Yeah, thank you guys for showing up. It's half of the journey just being here. So you guys are doing much better than how I started off with FTM. I did great, but like I was just like stress studying by myself. So it does get better. I promise you. Everything will start tying in when you hit MSK and CPR and everything. Like you'll understand why you're learning this. Okay, let's take a look at it. You guys were a bit split on this. It's okay. You guys haven't covered it in too much of depth. It's smooth muscle. So, and we're talking about dilations. What is your answer going to be? B.
Yeah,
Like
so
GMP.
we just went over it. We were talking about nitric oxide, CGMP. That's how you know it's guanylate, not adenylate. AMP, like cyclic AMP, is going to be associated with your adenylate cyclase. So when you guys do go over that lecture, I hope it's just kind of like adding in some details here and there, but hopefully this kind of gives you an overview of what it's going to be. And then here's the pathway again.
Exactly. This is pulled from your lecture material, so just be aware. Smooth muscle, cyclic GMP. Other stuff you're going to cover later on. I don't want to give you guys too much information now and get you guys overwhelmed. Okay, 6 answer choices. Timer starts now. You guys, make sure you click in. All righty, let's look at the answer. So it is GQ. Okay, it's a GQ inhibitory. Why? Take a look. So 10-year-old boy presents to ED respiratory distress after playing outside with sibling. Asthma is suspected. A drug is given that will decrease the amount of intracellular calcium released by the endoplasmic reticulum. What is likely the property of this drug? Remember, when you're thinking about lungs, we're talking about a different type of receptor, right? I don't know if you guys have covered it with your alpha, betas, and etc. But for the sake of time and your guys' sanity, it is a GQ associated one, okay? Next module, you'll come back for the exact same review and we'll tell you guys more about it in terms of drugs and pathologies associated with this, but it is GQ here, okay? Sarah, anything to add?
Nope. I think that should be good until you guys get to autonomics.
Yes. Then we'll have fun with actual receptors and names and remembering them. Okay. All righty. This is where I take over and do apoptosis and necrosis. This is prepping you guys for this week when you guys cover it. Now, they're going to spend this huge swath amount of time in one lecture going about apoptosis and necrosis. It's going to be like forever. It's going to feel like, but most of the things that they're going to test you on is going to be emphasized in these key principles. One, do you have a healthy cell? Hopefully you do, and not too many of them are going apoptosed, and not too many of them are, necrotizing. Okay, but in key cases, right? For example, during trauma, any sort of inflammation and swelling, necrosis can set in. That's where the cell swells, right? Due to its loss of its membrane composition, it's kind of being disrupted. So fluids and other particles float in and cause the cellular disruptions that you're going to see. Of course, eventually that's going to cause the cell to lyse, right? Lyse is just when it explodes, literally. And that is a key example of when necrosis, the actual physical symptoms of necrosis start presenting. You're going to see neutrophils, well, you guys haven't learned that, and lymphocytes all invade this local region, right? And in an effort to arrest the cell from spreading to others. Of course, it can't always happen fast enough. So you're going to see typically with this lysing or destruction, the necrosis spreads to other unaffected cells nearby. Okay. It has a nice ability to do that. Now, on the opposite, the flip side is apoptosis. Okay, this is a clean process. So the cell specifically shrinks first, and then it can start to fragment. It's going to form these blebs. Okay, if you hear the buzzword for blebs, that's going to tell you you're looking at some kind of apoptosis. Now, what's one cardinal example of apoptosis that happens? Feel free to unmute.
Program death for like tumors and stuff, and during mitosis, meiosis.
Exactly. Also, a great example are your fingers, right? Your digits. At one point, they used to be webbed. We were all fishies in the ocean. Unfortunately, or fortunately, we moved past it and we now apoptose everything in between in order to create your digits. So you can see someone with when their fingers are fused together, that's a failure of the apoptosis triggering system to kick in. Okay. So those are just your examples. Tumor is also a great one. Remember, with tumors, it can go both ways. It can go either to the necrotizing pathway or the apoptotic pathway. So just be aware of the key differentiating factors. Remember this: shrinking versus swelling, fragmentation versus lysing. Okay. These are called blebs. Okay. SU loves that word for some reason. They like to say blebbing, and then you should be like apoptosis. Cool. Feel free, if you guys have any questions on that, stop me. I can cover more on it in depth, but the hour is running past us. Next, you need to know the two different pathways for apoptosis, the intrinsic versus extrinsic. Now, being young doctors that you are going to be, what does the extrinsic pathway typically deal with? I know it's a little advanced. You guys haven't covered it yet. Just think about it and let me know.
Outside factors.
Outside factors, exactly. You're going to have like your natural killer cells, your cytotoxic T cells, all of them are assessing if your cell is yourself, as in the cells of your body, are either infected or aren't. So they can go in and activate a cascade of events. Whenever you think of apoptosis, start thinking of a cascade. But what are the key players of this cascade, right? For the extrinsic pathway, it's going to be very similar, but what you need to remember is porphyrin and granenzyme B, okay? Those two are coming from your extrinsic pathway. Obviously, they're going to join with the other initiator of the caspases. The key ones to remember are 8, 9, and 10, okay? Those are the common ones. that you need to or will likely be tested on. The other one is the executor one. Okay, these are the execution Cas spaces. The important one that SU will likely test you on is 3. Okay, if you hear Cas space 3, that's the execution one. What that will do is basically tell the cell to pop all of the lysosomes that are there and start just degrading everything. Okay, that is your extrinsic pathway. Now, intrinsically, this can happen too. How can it happen? Here's a key example. You have DNA damage, right? You are a bad tumor cell, bad, okay? What it's gonna do is cause the P53 pathway. Now, you guys know P53, you've seen P53 before when they started talking to you guys about cells. P53 is one of the critical regulators and modulators of your gene integrity. It's gonna say, hey, there's irreversible damage. It's going to upregulate BAC, BAX, and BAD. If you see these, then you know that you're talking about the intrinsic pathway. Now, there's pro-life portions of the cells that are going to be constantly trying to make sure that the cell doesn't apoptose, and that is your BCL2. Okay, BCL2 gets downregulated by P53, and BAX and BAC and BAD get upregulated. It's going to help it maintain the next series of cascades, which is going to basically go in to the mitochondria and create holes, right? What is the in-between part of the mitochondria? Not the cristae or the inner region. What is the in-between region called?
Intermembrane space.
Intermembrane space. Exactly. The intermembrane space has cytochrome C. Cytochrome C is very critical. If you it's constantly held in the cytochrome C, or sorry, in the intermembranous space, and cytochrome C is trying to desperately get out. BAX, BAC, and BAD are kind of like your get out of jail free card for cytochrome C. They basically create pores for cytochrome C to pop out and then wreak havoc with the APAF1. Okay, APAF, they're gonna get together like two bad bandits and create the apoptosome. Okay, apoptosome is going to once again create this complex. That complex is going to merge with and initiate 8, 9, 10. You don't need to know too much about 2. It's 8, 9, 10. And of course, when you're about to execute something, it's 3. Okay. Those are your intrinsic versus extrinsic. That's the short version of what SGU is going to spend an entire day going over on this topic. They can only test you up to that level because you guys won't cover the clinical, super clinical components of it, right? That's what we saw in our exam. That's what you'll likely see as well. Lovely. Let me clear this before I move on. Next one. Have you guys covered this initiator, elongation, and termination?
Yeah.
Awesome. Okay. Initiation, creating the pre-initiation complex, right? Those are your RNA polymerase and all of the things to unzip the helicase and all of those, genes opening portions are critical. Okay. Those you can memorize. Those are all from undergrad knowledge. I don't want to spend too much time going over. Obviously, they're going to unwind the DNA, and they're going to start transcription. Now, what is the cardinal rule for this? Is that any genetic information that they present to you is presented from 5' to 3'. Okay. Even if they don't label it on the side of 5 and 3, always remember that 5 to 3. Okay. Because we had a question on this, and I'm pretty sure some folks got it wrong because they were super upset that they didn't label the sides. Okay. Then you have elongation, which is elongation of the RNA strand of creating the actual RNA template. Okay. And then finally, it's your mRNA, but it's not your mature mRNA. What do we have before that? What's the distinguishing factor between your mRNA and your mature mRNA?
Transcriptional modifications, polytail, and GCAP, and transcription.
Exactly. You do have to know that, guys. please, I will tell you, those are the points that slipped away from some folks because they forgot to memorize elongation factors here and poly A tail there, because they tie into you creating the composition for everything that you're going to be coming out of the cells, right? That's going to be coming out of the cells. Okay. What's the quick mnemonic for remembering your prokaryotic ribosome complex versus the eukaryotic ribosome complex?
Okay, our eukaryotic is much larger.
Eukaryotic, yes.
Mm-hmm.
But the quick way to remember your subunits.
50 plus 30 is prime, and 60 plus 40 is even.
Exactly. That's the quick way to remember the numbers and not mess up too much, okay, on getting your prokaryotes versus eukaryotes wrong in terms of the ribosome complex. Now, they create ADS, right? Even number. So what else do you need to know? Now, where is the wobble region in this portion? Is it in the first position or third position?
Third position.
It is the first position in terms of the tRNA. That is the trigger right there. We were given one of those questions on the quiz about it, and they asked us, in the terms of tRNA, where is your wobble region? And I was like, yes, it is 100% the third position, but it wasn't because they were talking about tRNA. Okay, tRNA, it's in the first position. Okay. Now, wobble region, what does that allow for?
Coding of different proteins.
Coding of different proteins, genetic diversity, yet again, of how those proteins get made. And obviously, if you change a amino acid sequence that's created based off of the wobble region, it's going to have downstream effects of how the protein gets made or how the protein shapes and folds. Okay. You do need to remember the site of amino acid attachment. I saw a question on that. That portion and how it's loaded is very critical with the phosphate being there. It basically stabilizes this whole complex and allows for the amino acid to be retained here. And then your anticodon loops, you guys know that. Remember how it's numbered, it's always 5' to 3', right? So they might give you an anticodon loop of I, C, G, but you have to remember the codon is 5 to 3. So your wobble region is actually in the first position. Okay. That's the trigger right there. Awesome. Let's look at the next thing. I just put this in because I didn't want to overwhelm you guys from all the material from the lecture, but remember, you do need to know your elongation factors. You need to know that you use GTP when you attach to various slots, right? What's the mnemonic? APE, right? APE is like P is the kind of the start site when you're doing it. That's the initiation portion. Now, after you do the first initiation portion, everything else is going to follow along with this process of APE, right? It's A, P, and then exit E. Okay, that's just a short form of it, but please spend some time going over the particular enzymes. I don't want to burn you guys out too much. Okay. Next one. This is how everything gets made, right? This is just a short, nice picture to represent it all. Go through each of them, especially know that the SRP region identifies the signaling peptide, okay? And it's going to be pulled in and then allows for things to further get modified. Now, what do we call this, guys? What is this organelle and what do we do here?
Is it rough ER?
It's rough ER and or what do we do in the ER?
You have a signaling sequence, and then you pretty much bring them to. They have the signaling sequence for rough ER, so you like a zip code to deliver it to rough ER.
Exactly. And in the lumen of the ER, you are also going to do post-translational modifications. Okay. post-translational modifications happen in the ER. Remember, okay, you guys think back to your initial introduction to your organelles. What tag do we add to something that's about to be degraded?
Ubiquitin.
Ubiquitin.
Exactly. But the lysosomal, that's ubiquitin ubiquitin proteasome. But if we're going to send it to the lysosomal pathway, what's the tag?
Mono 6-phosphate.
Mono 6-phosphate. Key thing to remember is that the mono 6-phosphate is added in the ER. Okay, if it's happening here in terms of modification, it's happening in the ER. Of course, there is some modification that happens in the Golgi, right? Sorry, ER. This is the Golgi. I'm all over the place a little bit here, but remember, it happens in ER, and then you have the Golgi that's doing the assortment. Remember, what does COP1 do?
Retrograde.
Retrograde.
Retrograde. What about COP2?
Interrograde.
Interrograde. Okay. One is going out into the world, and the other one is bringing it back in towards the nuclear side. Okay. Now, what is this portion of the Golgi?
The cis.
That is the Golgi. That is the cis. What's the other one?
Trans.
Trans. Okay. What about this one? What is being attached to the side of a transporter?
Catherine.
Catherine. Exactly.
Yeah.
Awesome. Nicely done, guys. Now, why do we need post-translational modification? Why on earth are we modifying things further? Good question. I don't know. But in all honesty, you do need post-translational modification because most, if not all, of the proteins that you create or peptide sequences need to be tailored towards their specific goal of either acting as a receptor or serving as a receptor itself. One great example that you guys will cover or have covered is your insulin. Okay, endogenous insulin comes in various shapes and forms, depending on where you are in terms of the modification process. Pre-proinsulin folding happens in the ER. Okay, RER specifically. Proinsulin processing is happening at the Golgi, right? It's at this point we eject C-peptide. Okay. Now, C-peptide, I don't know if you guys have covered it. What is that a good marker for?
Half-life for insulin.
Half-life for insulin, right? It's a great example. Even if you can calculate hemoglobin A1C, you can use C-peptide to understand how much insulin is floating around in someone's body, okay? And finally, when you do eject it, the actual insulin, like the functional component, that's happening in the vesicular point, right? Like the vesicles are popping out these wonderful insulins that's keeping us alive, not going into hyperglycemic shock. Awesome. You guys have covered prions. This was a minor point, but we did get tested on it, and a lot of folks forgotten about what a prion is. What is a prion, guys?
Misfolded protein.
Misfolded protein. Of course, a prion itself, we have endogenous kind of prions inside of us, but when they get changed from the alpha structure, non-infectious PRP state, they turn into the beta sheets, right? The beta sheets that you're seeing there, that becomes a more deadly version, right? What's an example of a prion disease?
Kuru.
Mad cow disease.
Kuru, mad cow, those are all good answer. Now, what is not able to destroy this?
Proteasome.
Proteasome, ubiquitin proteasome pathway. It's a minor point, but I swear I had a question on it, and I only got it through because I was able to. I literally looked at this slide before I took the exam. Okay, lovely. Now, a final thing. You guys have learned a lot, right? You guys have learned so much things, starting from the genetic level to the RNA and the protein level. But you have to make sure you tie in the drugs for each of the location, because it is a minor point, but it's going to be helping you get those couple of extra points that some folks might not be able to catch here and there. Now, at the DNA level, know these drugs, okay? I put a list of them. I made it simple. Whiteboard these out and associate each of them to their specific components, right? DNA to RNA, what's getting blocked in the process based off of drug, rifampin, amanita, both of these are blocking that process, right? Via RNA polymerase or inhibiting RNA polymerase 2, okay? The RNA level, there's the initiation, elongation, and termination factors, right? The only one that drugs of critical importance is like streptomycin, tetracycline, erythromycin, chloramphenicol, cyclohexamine, diphtheria, you've seen diphtheria before, and the puromycin, okay? And they're gonna cover diphtheria in a different portion of your lecture, I think this week. Okay, anti-cancer, you guys have seen these, hopefully, in the first lecture. Colchine, Taxol, Paloidins, these are from those mushrooms, death cap mushrooms, don't forget. Amanita, Paloidin, that's two different things it's acting on, so don't try those fun mushrooms in the forest. And then the cytocollisions, which block the actin polymerizing and blocks the cell movement, okay? And then protein level, what you're happy, go for it.
Is colchine different than colchicine? Or is it a type of?
Colchine is different than colchicine.
Okay.
Yeah. Now, there are drugs that act at the protein level. Acetosolamide can cause acidosis, Prozac, serotonin, DFP, poison, acetylcholine esterase. You'll see more about that in FTM2. Aspirin, caffeine, know how it's increasing cyclic AMP is something you'll cover in your G protein system. Allopurinol, you guys remember your HGPRT and leishnion and all of those things. Allopurinol is typically used to treat this as well. There's also cholera and E. coli, pertussis toxin. All of these, I highly, highly suggest you guys whiteboard this multiple times so you have a mental map during the exam that you can draw upon and say, oh, I need to give someone ciprofloxacin. It seems to be an antibiotic. Where is it acting? On the DNA level rather than on, a different level. Okay.
Just want to say
All right.
that colchicine is the drug. There isn't something called colchine, so that must have just been a misspelling. Mistake.
Yeah, colchicine is the one that when we were talking about your fish, it was the one that arrests it, I believe, in metaphase. Or no, I'm sorry, that was for the G-band karyotyping.
Yeah.
Yeah.
Did I misspell it?
I don't know. I don't know how to spell any of this
Called
stuff. That's fine.
colchicine.
Yeah.
Okay. Let's do questions, guys. As more practice, the merrier. We have a couple for you guys. Let's get this started.
We totally understand that you guys are probably exhausted by now, so we will give you guys less time for these questions so we can kind of run through them quicker.
Make sure you click in. A few seconds remaining. Right. Let's see how we did. And the answer is C, as the majority of you guys got. Remember the drug, right? The mechanism of action for erythromycin is it binds to the 50S subunit and doesn't allow for you to create the functional subunit, okay? And here is the summary slide from your lecture. So you guys can take a look at that. I'm not gonna spend too much time on it. Let's do this one. Quick answers, guys. Hopefully I'm not giving you guys too long of a vignette. A couple of more seconds. Feel free to click in. All righty. You guys are absolutely correct. It is C-peptide. It's a great marker instead of using oh man, I'm losing my words. Hemoglobin A1C. There we go. Yikes. It's been a long day. You guys can tell. Let's do the next one. There's the picture. There's the folding process. This is pulled directly from your lectures, by the way. So feel free to take a peek after. Let's do this one. Okay, we had a bit of a split. The answer is D, okay? Now, the quick way I remember of how actinomycin works is act in the middle is how I remember it. So intercalates between the bases and the DNA and basically creates a kind of a stopgap. It doesn't allow for transcription to progress. Okay, that's the answer. And next one. Make sure you guys click in, a couple of more seconds. All righty, so how does ciprofloxacin work, guys?
Inhibits the DNA gyrase or topoisomerase.
DNA gyrase. Ciprofloxacin, yes. So it's topoisomerase, right? Because DNA gyrase is for who? For, is it prokaryotes or eukaryotes?
Prokaryotes.
I think it's DNA gyrase because it is prokaryotes.
Yep. Awesome, you guys got it right. Ciprofloxacin.
More on one than on the other, right? It affects prokaryotes more than the eukaryote.
Correct. Right. But in high levels, it can have negative effects. Okay. And awesome. Let's do this one. Give me 1 second. Opening and polling again. These are the type of questions that they can ask, okay? So like they're never gonna ask you like, what does this particular organelle do? It's always like, here's a pathology. Now tie it back to the foundations of medicine that you guys have learned. Which organelle is likely doing this? And then they'll ask you that question. Decreasing the timer so that you guys can quickly put your answers in. Alrighty, let's see how we did. The answer is C, guys. What organelle are we talking about?
Lysosome disease. It's a lysosome. It's lysosome proximal disease.
Yeah, it's a peroxisome disease, exactly. So that's the quickest way to know that Zwelliger deals with peroxisomal disease, or sorry, peroxisome organelle level diseases, and it's typically involved in metabolism of very long chains of fatty acids, also involved in the alcohol degradation pathway. Just be aware of that too, just for good measures. That's the answer. Cool. Let's do this one. Ooh, okay. We have a split. Okay. So, Sarah, do you want to take this one?
I honestly didn't even read the question, but
No, no, no. If
you
you
want to
want,
go ahead?
it's up to you because I was like, Ube,
I just
the
got
role
back.
is yours.
I had to go do something. Sorry.
No worries. Okay. So a 4-year-old child is seen by the doctors found to have a mild intellectual disabilities with rapid weight gain, with a huge appetite. Prader-Willi is the diagnosis. The diagnostic analysis of the child will likely reveal what? So the answer is B, okay? Inability to produce snurpin. Now, is snurpin paternal driven or maternal?
Paternal.
Paternal.
Paternal, exactly. Now, what is likely like the genetic mechanism? It's a bit of a tough one, right? The paternal imprinting of snurpin. Do we have paternal imprinting of the snurpin, guys?
No.
No, exactly. So the maternal copy of snurpin is the one that gets imprinted on. Now, imprinted, like just to reemphasize the point, is the methylation of that equivalent gene, right? The dad's giving you a copy of snurpin, the mom's giving you a copy of snurpin, but remember, the mother's copy of it is imprinted on, AKA it's silenced, okay? Methylation, silencing, equivalent languages. But here, what will likely involve is the deletion of the paternal gene of snurpin, whether it's a deletion or a mutation, anything of that equivalent sort is just telling you what's happening. So the answer in best of worlds is B, okay? Obviously, they can ask you more details in the exam, but any questions?
Quick question. So the word imprinting in general, it doesn't like imply a disease. That's just normally what happens.
Correct, exactly. Remember, our genome is huge, right? There are portions that have to be methylated in order to not allow for everything to be expressed all at once. Imprinting is a specific form that's associated with the copies that you get, right, of Snurpin, Prader-Willi, etc., based off of it, whether it's Snurpin or Ube. Now, typically, when we're looking at Snurpin, it's going to be basically dependent on if you're talking about the maternal or the paternal copy, because the maternal copy is going to be imprinted, whereas the paternal copy is the functional one, the one that's actually doing the function of creating the Snurpin product.
So they have to be autosomal?
Do they have to be autosomal? You guys want to?
We're talking specifically about Prader-Willi and Angelman. So you're not applying this to other disorders because we're talking specifically about a certain gene that is naturally methylated in one copy and not in the other. So I wouldn't try to apply this principle to the other like autosomal recessive, autosomal dominant topics.
Quick question. If option C said duplication of the maternal UBE3A gene, would that be the correct answer or no?
No, so when you're talking about these disorders, you're not getting two copies. You're either having your one functional copy or you lose that one. So you're not going to get
What
duplication.
happens in uniparental disomy then?
Uniparental disomy is when you have only one copy, I believe, and it happens to be the non-functional one.
Okay.
So for this one, it would be uniparental disomy of the mom.
Uniparental disomy, I'm not 100% sure for this one. I can check and get
Because
back.
they said that it could happen. Okay, because they said that it could happen two ways. One where the dads would be silenced and it just wouldn't be there, or basically would be getting two of the moms. So that would be the uniparental disomy. And then if the dad is silenced, would that be maternal imprinting of UBE3A?
Yes, I think.
So if B wasn't an answer in this and E said maternal imprinting of the UBE3 gene, then that would be correct?
Yeah, but the only thing is when you're talking about maternal imprinting, that's normally what happens. So like you would just be saying like, oh, chromosome 15 is maternally imprinted. Yes, that's correct. But when we're talking about causing Prader-Willi or causing Angelman, something has to happen to the other copy.
Yep.
Okay.
Remember, there's maternal. Okay, but there's uniparental disomy. That's you're talking about in the germline where you have a kind of a rescue system, right? Where you're getting two copies in order to have fetal viability for it, like for everything to continue to grow and develop. Obviously, they're going to be missing something because they're only getting two methylated copies of the maternal stuff or the two methylated copies of the paternal stuff. Okay, that's uniparental disomy. Just be aware of that in terms of definition. Let's do this one. Make sure you guys click in. you're probably wondering why you're getting questions from early on in this block or module. It's important that you guys all constantly do a cumulative review of the material, right? From week 1, week 2, etc. So just so that it's always fresh in your mind before the exam. The answer, you guys were a bit split on this because I can see how you guys were having difficulty with this. Now they're having what is happening, right? It's lower part of the esophagus presents with simple columnar epithelium with goblet cells, which the following types of cell adaptation is best described here. It's metaplasia, right? It's metaplasia. Now, if you were to diagnose this, it's probably GERD, GERD, right? But in this case, they just asked you in terms of purely definitional, what is happening here? It's metaplasia. Now, is metaplasia reversible or irreversible, guys?
I don't think we've learned this before.
Yeah, I don't think we did this yet.
Cell adaptation, I think, comes next week.
Got it. Cool. Little advanced. You guys will have these slides anyway, so make sure you guys review this when time comes. Thank you guys for letting me know about that. Awesome. Let's do the next one. Make sure you guys click in. Alrighty. So it's Duchenne muscular dystrophy, and you guys now know it's X-linked recessive disorder. And the answer, whenever you're thinking about DMD, it's going to be frame shift. And they're going to talk to you guys later about this too. So just be aware it's due to a frame shift mutation, but they're going to give you different types of this. Okay. Awesome. You guys did great. Go for it.
How do you know that it's Duchenne and not Becker? Or do you,
DMD.
aren't they both?
Duchenne. Becker's muscular dystrophy is part of. Can one of you guys help me out with that one?
They're the same. It's just Becker's is less severe than Duchenne's.
Becker is an in-frame mutation and Duchenne is a frame shift mutation. So like anything that's a frame shift is always going to be worse. I always had to study these before exams though, because we would get them mixed up. But Duchenne is frame shift, Becker is not.
But I mean, like based on the like the paragraph, how would you know that it's Duchenne in order to pick frame shift?
So the waddling gait thing, I'm pretty sure is a buzzword for that, but also with these, just look at the age, because if it's a really young kid, it's usually going to be Duchenne. And if it's a person that made it to adulthood before they started having symptoms, then it's going to be Becker because it's less severe.
Thank you.
But the questions on the exam will definitely be more clear in determining like if it's Duchenne or if it's Becker. Because you're right, yeah, they're both the same gene.
Yeah, I think, sorry guys, I was confused between that and myotonic dystrophy. But thank you guys. Let's do this one. Okay, I'm gonna slowly start winding down. You guys are clicking in. Alrighty. So the answer in this case is hemosiderin, okay? Make sure you guys look at this, okay? We did not look at it, and there was at least 1 or 2 questions we were like, oh shoot, we could have gotten that so easily. If we had just taken a quick peek at that page where it was in our lecture about what each of these look like, okay? The answer here is C.
If you guys do have access to our Facebook page, I believe our my OPLG slides, I did go over all of these and like what they look like on histology slides. So if you guys do need like more clarification on it, just go look at that. It's on our drive.
Which drive?
The Sparkles DES, sorry.
Okay, thank you.
Yeah.
Quick responses, you guys know this. We went over it. I have full faith. We're looking for 100%. Alrighty, let's see how we did. And the answer is D, COP2, right? This is anterograde. It's moving from the ER to the Golgi. It's not going back. Going back is 1. Okay. Let's do next one. This is a nice test of how much you guys kind of took a peek at all of the definitions. I swear I even got one of these from their questions back in the day. Almost done, guys. You guys can do it. This is stamina, right? I always tell you guys it's a marathon, not a sprint. And the answer is A. Okay, variable expressivity, right? Let's take a peek at this. It's 2 members have split feet, 3 of them have split hands, 6 have a little bit of both, right? That 6 had a little bit of both is the variable expressivity part that should clue you in. Excellent.
Would it also be D as well because it's affecting the same gene from the same location?
I could see that. Anyone have issues with it being both?
I was thinking it was both too because it's only describing one gene, so different expressivity of the same gene.
With your allelic heterogeneity, it's going to be the same gene. It's going to be different mutations on the same gene. So when you're talking about people in the same family that have the same exact mutation, then that's not considered allelic heterogeneity. They just have the same disorder with the same mutation. But you would have to say like, oh, this person has TP63, the other person has like some different mutation that's also on the same gene location. Does that make sense?
It made sense to me. Awesome.
Makes sense.
Yeah.
Okay, so let's talk about this. Is this what are we seeing here? You guys are correct. The majority of you guys got to the right answer. It
Autosomal
is
dominant.
autosomal dominant, exactly. So a great example of that is FH, which is familial hypercholesterolemia. Okay, you're going to learn more about that when you move into CPR. But for the time being, just know that it's autosomal dominant and it has a nice vertical inheritance pattern, right? As all autosomal dominant hopefully should. Let's do this one. It's a bit of a long one. I'll give you guys some more time.
I have a quick question for the previous one. What are the commas under the thing?
Oh, yes. So that goes in order, right? Of like where they are. Or you're talking about like these commas, right? In between.
Yeah.
I
I thought there was an
believe
example for like Huntington's where like it would like represented repeats and how like the next generation got more, but this is clearly not that. So I'm not sure what this is.
I remember specifically Dr. Sobering using this as like generational things. He was like, generation one is like in this position. So this might have just been mislabeled because I did pull this from the web.
Oh, okay.
Let's go back. Let me open the polling. And don't worry, guys. I will make sure that I upload post review. I'll upload some practice tests and examples for you guys to look over. But make sure you guys do the Sakai questions, okay? The Sakai questions are a great benefit. Okay, so majority of you got it. It is E. NaCl concentration does not change, right? It's important to remember that. Why is it not changing, guys?
Because it doesn't cross.
Exactly, it's impermeable. So the concentration in the bathing solution will not change. So the glucose concentration is penetrable. So it was going to go right through and it's going to change the internal composition, right? This is once again going back to the principles that you guys learned with the osmolarity and tonicity, right? What is a transient change and what is a more permanent change? Okay. Next one. And I've been told that next time I'm gonna give you guys a break in between so that you guys aren't so wiped out. We'll pull in the nice trots in between. You enjoyed this 30-second break? Have you guys had Dr. Trots yet?
Yes, we have.
Yes.
Isn't she the best?
Yeah, she's so cute. She's adorable.
She's great. We have a lot more of her during term 2. So remember, that's motivation for you guys to keep going, get through it so you can have trots all the time. All right, we have a nice split on this one. So, okay, guys, describes a steroid. What does a steroid do? Can it cross the membrane?
Yes.
Exactly. So the answer is it's going to affect the gene expression directly. So there are steroid sites, steroid binding sites on the genetic portions in the nucleus. So it can go directly in there, slot itself there, and change your expressions. Okay. Awesome. let's do this one. Almost close to the end. And 5 exclusive. And let's take a look at it. So the answer is Golgi. Exactly. Now, I know you guys were probably tempted by ER. So just be aware of where your pro-insulin, whereas your C-peptide, your pre-pro insulin, where that is located versus when your C-peptide gets released. C-peptide gets released in the Golgi process, and then eventually you get the final product being released as a vessel. Okay, awesome. Let's make this the last question and then we will call it. There are other questions below, but we can end it a little early since I've given you guys a boatload of questions. All righty, let's look at the answer. Okay. You guys were a little bit mixed on this, but most of you guys came to the right answer. So the answer is D, right? The cell transiently swells, right? And then returns to its original volume. That's really due to one important thing, right? NaCl doesn't cross, but your 20 millimolars of urea can cross, so that equilibrates it to a level. So final structure is going to be a-okay. It's going to be isotonic. Okay, let me quickly end it and say thank you guys for attending. Any questions? I can stay for another like 5-10 minutes and answer any questions, but I'm gonna stop recording right now.